Good morning, one and all. Am I audible, Akash?
Good morning, sir.
Good morning, good
You
morning.
you audible, sir.
So I think near about 48 participants have joined today. So shall we start now? PPT is visible, Akash?
Yes,
Okay. So it is in slide mode. It is okay, Akash?
Yes, sir.
Okay. So friends, yesterday we have seen the different topics which are related to the sustainable production of grape. So one of the important topic is that stress, particularly abiotic stress management for sustainable grape production. So as we know that the stress Levitt, which is also known for the father of plant physiological disorders, he defined that there is a change in environmental conditions that might reduce or adverse effect on its plant growth and development. So we can say that in case of abiotic stress, rather than biotic factors which causes adverse effect on plant growth as well as its development, we can say that it is a abiotic stress. So the types of the stresses are there. As I told you, a biotic and a biotic. So biotic, where we know that it is due to the microbials and other biological means. Particularly in a biotic factors, mainly there are 3 to 4 factors which are responsible for abiotic stress. That is water, whether excess water or deficient. It may involve your rainfall or hailstorm also. Second, temperature, high, low, or it might be the cold. Salty conditions, edaphic factor or more ionization is there. So probably there might be toxicity or deficiency of the particular nutrients or salts. They may cause abiotic stress. And last is the air pollution. So as we know that the grape is very important crop. Actually, it's a temperate crop, but giving more response in a tropical condition also. So in this lecture, we will see that how the abiotic stresses affect the grape cultivations. Definitely, there is a reduction in the yield and quality. I just told that there is a reduction. It means that if we'll see the complete plant process, physiological process of the grape vine, right from its sprouting, flowering, fruiting, horizon, all stages will get affected due to the abiotic stresses, so that there will be reduction in the yield and quality. And due to that, there will be huge economical losses also. And third is that ecosystem services. Probably most of the participants know about the ecosystem services. The services which are given by the plants itself may be any agronomical crop, horticultural crop, or forest crop, even the grasses also. So with concern, in case of the grapes, we can say that due to the reduction in the area of the cultivation as the effect of abiotic stresses, it may adversely affect your biodiversity support. What biodiversity support? If anybody having the orchards, vineyards, there are beneficial organisms like your honeybees, like birds, so which are maintaining little bit biodiversity in the in that particularly vineyards. So it may get affected. Definitely the carbon sequestration. Each and every grapevine sequestrate the carbon through their physiological system, and through root system it will get sequestrated in the soil. Definitely the water filtration, yes, water filtration through the root zone, it is also affected. And last is aesthetic and recreational value. As we know that it's a very impressive crop. Anybody can attract due to that the plant, we can say the leaf structures, then the plant look, vines, and have the bunch load and all these things, different colors. So most of the peoples are using the grape plantation as creating a recreational value, like most of the restaurants where the nearby grape plantation is there. Sometimes small cafes are there. So all these things are related with the ecosystem services. They are also affected due to the abiotic stresses. If we'll see how the abiotic stresses affecting the grape last from your 2001 to up to 2023-22. So little bit there is a increase in production, but sometimes there is a decrease in production, particularly in 2009 and 2010 year. But simultaneously, there is increase in the area, but sometimes there is decrease in the production. So here we can say the red line shows that sometimes due to the abiotic stresses, maybe erratic rainfall, hailstorm, rise in the temperatures, all these factors leading to a decrease in the production. So here we can see in this graph. As I told you that the grape is very important crops, and 2 times pruning is done, particularly in the tropical condition. So particularly in the foundation pruning, it is done in the March and April. So it affects most of their physiological processes. Which factors affects or which are very important, particularly high temperature during your March and April time. Sometimes there may be the water deficit conditions. More irrigation is also required due to the high evapotranspiration rate. And sometimes there might be the onset of rainfall. So they affects the your foundation pruning and onward practices, particularly the plant growing process, right? Your sprouting, flowering, all this will get affected. Same case of fruit pruning also. Unseasonal rains may be there in month of September or October. Sudden fluctuations in the temperature may be high or low. So all these factors affecting both kind of the pruning, whether it is the foundation or fruit pruning, which is related with the grape vine. Here we can see that, as in last slide, I told that the most of the important relevant factors affecting the grapevine different growth phases, right from pruning, sprouting, then your 2 to 3 leaf green stage. After that, simultaneously, there will be the parent green stage, means bunches will come out from the skin, then flowering, then fruit setting, up to the ripening. All these stages may get affected due to the abiotic factors, as I mentioned to you. So here we can see that at a global level, particularly India here, it comes under the severe water stress conditions, probably in from 2020, 2030 to 2050. There may be chances of getting the severe water stress. So as of now that there is increase in the 50% production, but onwards up to the 2050, your production may get low down. It is predicted by the OECD environmental outlooks up to the 2020, 2050. Similarly, here we can see that vulnerability of the climate change, particularly in India. So here we can say that very high vulnerability has been also shown, particularly in most part of the Maharashtra and sometimes some part of the Karnataka also, where the more than 75 to 80% grape belts are there. So it is one, we can say that it is one kind of indications that probably this area will affect more in future also. So more than the 90% simulations predict that there may be increase in the drought conditions, particularly in the subtropics to the end of 21st century, while there will be increase in the extremes in precipitations as predicted in the major agricultural production areas of southern and eastern Asia, eastern Australia, and northern Europe also. So yesterday, you might have seen that in a lecture of the irrigation management, there is a high demand of water requirement of the grape vine. So considering the crop growth stages as well as amount of the rainfall and evaporative demands, so the water availability and quantity of water, which is very relevant for the grape production. So here we can say that near about if recommended irrigation schedule is there, so 45 lakh liter per hectare water requirement for one season, which is for the recommended in the grape. So most of the farmers are using more irrigations. They are not going to follow the recommended irrigation. So ultimately, there is a loss of the water as well as water use efficiency also low down, and will get also their it will also get affected. So recommended irrigation schedule is very important in case of grape. If we see the view of the future where may have the probability get reduction in rainfall or probability water stress might be there. So as study were done in the NRC grape, according to the varieties, most of the varieties show the less water requirement, particularly Fantasy seedless having the less water requirement compared to Thompson seedless. So it has been observed in this study. So likewise in wine grape varieties also, particularly specific varieties according to their size and shape. So it is, we can say that varieties also require the different quantity of the waters, and they also require the particular schedule. So we know that IPCC, Intergovernmental Panel on Climate Change, it is governed by the UN agency, and they predicts that there might be most prominent challenges, particularly in grape crop in arid and semi-arid conditions. So there may be chance of abiotic stress, particularly drought, in upcoming futures. So it is now we can see that particularly in your 2022 and 2023 probability of there is a decrease in rainfall, and we saw that there is a rainfall but might be more erratic rainfall. At the time there might be more rainfall, but in there may be a frequent discontinuation in a whole season, so which affects the growth and other stages of plant, particularly of grape plant. Here we can say that if moisture deficiency is there, in particularly foundation pruning and fruit pruning, so there is a chance of getting the yield reduction. So here data shows that if 50% reduction in irrigation at the shoot stage may reduce the yield. Similarly, in fruit pruning season also, so irrigation schedule is very important, which may save the water and which will be helpful for increasing the more area under the grape cultivation for the stakeholders. So what should be the solutions over the drought, or we can say the moisture stress? So we have to use subsurface irrigation, or we have to follow the recommended schedule of irrigation. We can use the mulch as well as anti-transparents, as or we can go for the partial root zone drying also, so which will help to save the water, more water in a grape cultivation. We can use the mulch as well as the anti-transparents. I think in yesterday in last lecture, Dr. Yukti Verma has already mentioned about all these things. So I will skip. So mulch plus anti-transference will be really helpful, particularly your cost-benefit ratio with respect to your yield also. Subsurface irrigation is very important for grapes, so it can be followed, and we can save the water up to the 40 to 47%. So as we can see in this slide, cost-benefit ratio and your yield as compared to recommended irrigation also, somewhat subsurface irrigation is more important and shows the good response over the yield. Same we can see here in this slide. So the comparison between surface irrigation, recommended irrigation, and partial root zone drying also. So it can be followed for saving the water. Same time, with different growth stages. So NRCG has given already schedules and farmer practices. So we can see that due to the recommended schedule, there might be water use efficiency will be more, and farmers may get a good yield also compared to their regular schedule. So decision support system is also very important. So how farmers have to decide at what stage how much water or other things to be given to the vines. So that's why decision support system is very important for them. It is very informative. So farmer have to download this particular app, and they have to put their data in through that software, and they will get the further guidelines that at what stage how much water and other practices can be done, particularly in the grapevine. And it has been developed by the ICAR, so it is very important for stakeholders. So next, abiotic stress factor, that is the soil salinity. So we know that Maharashtra is very much area under the grape cultivation in Maharashtra. So soil salinity problem is also there. Akash, am I audible? Okay. So this is also one of the factor which affecting the grape cultivation. So we will see that. So what are the more source of the major source of soil salinity, particularly the water? Sometimes whatever the fertilizers which are given to the plants, that is also source of the soil salinity, particularly the salts. So tube well water, bore well water, as well as some canal water, they are also they are also a factor which may increase the soil salinity. This is one of the important thing for taking considering the soil salinity problems. So here we can see that in data. So how we can escape the soil salinity issue, or how we can tolerate that problem if anybody have this soil salinity little bit issue. So we can grow the different kind of the rootstocks, particularly recommended rootstocks. And study has been conducted in NRC grape with the different rootstocks. So here we can see that 110R, which shows the good response toward the soil salinity, particularly as we know that it is exclusive to the sodium salts as compared to other rootstocks. So and followed by the doggeries. So these rootstocks can be utilized to avoid the soil salinity problem. So here we can say that an early onset monsoon is as well as the cloudy weather condition during the different growth stages. So this kind of the climate which reduces the fruitfulness and flowering in the grapes. So I think most of the more than the 57 vineyards which have been observed, and the data were recorded, and it shows that definitely there is a huge effect on its fruitfulness. So very important thing regarding the temperature. So temperature, whether high or low, is also causes it also causes your reduction in the yield and quality of the grape wine as an abiotic factors. So there might be shifting in phenological stages. So as I in earlier slides, I told you that there are the different phases, right from your foundation pruning to fruit pruning. So there is change in temperature. So in the yesterday, someone has asked the question whether we can grow the grapes in other belts rather than Maharashtra, Karnataka, or Tamil Nadu in the India. So I mentioned that growing degree days, or we can say the heat units, are very important. So if there is a rise in the temperature, particularly 1 degree Celsius, so it adds near about more than 200 growing degree days, or we can say the heat units. So due to that, your growth phases of particular grape vines, they may get affected, and it will have it will show effect on your yield as well as quality. So due to the temperature, your quality may get reduced, and other things related to your production, particularly in related to your table grapes or wine grapes. So it will get hampered, it will get affected. So temperature is also one of the leading factors which affects the growth as well as quality in the grape. So here we can say that if there is a rise in temperature, particularly in different growth stages, right from your sprouting and to cane maturity or other growth stages. So leaf scorching, or we can say leaf drying, it may happen there. Sometimes there may be a partial maturity of canes, even though particular days which are required for maturity has been over, but it may show there is a little bit immaturity in cane. So if there is a immaturity in the canes, so there will be not proper fruit bud differentiation. If not proper fruit bud differentiation there, then you will not get the bunches, and it will indirectly affects your yield for per unit area. So temperature is very important factors, which is in relation to the vegetative growth phases of grapevine. Here we can see that how the temperature affects the sprouting. It is one of the phase of the grapes. So the experimentation has been carried out that we have used the green net to cover the to cover the proportion of the grapevines, and it shows that there is a good sprouting as compared to your open vineyards, open condition of the vines. So these practices that can be done. So use of the shade net, that probably helpful in the region where the fluctuations in the temperature, particularly in the after the foundation pruning and fruit pruning. So temperature may affect here. We can see that particularly after fruit pruning, berry shriveling that can happen. Similarly, sun scalding, it is also there, and discoloration in the berry color, it may also happen. So as we know that if temperature may lower down, so there may be probability of other physiological disorders also, like the pink berry formation in Thompson seedless. So these kind of the things that can be seen due to the fluctuations in the temperature, whether high or low. So here we can say that occurrence of phenological stages as affected by the temperature variation in the time of the pruning. So here the pruning were taken in early April, and this it is in early late April. So we can see the complete difference that if sudden rise in the temperature, which may affect the sprouting. So this is the pictorial depiction of that things. Here we can say that after the foundation pruning, so there is the initiations of the sprouting, and at a different 10 leaves, 7 leaves, 3 leaves to 10 leaves. So it is also affected due to the rise in the temperature. So experimentations were carried out with in a foundation pruning and fruit pruning at the same time of the foundation pruning, and but there is a change in the date of the fruit pruning, and it shows the little bit difference. So we can say that here in P2, that is April pruning, and probably your foundation pruning is after 15 October. So it shows that there is a more yield as compared to your late pruning, foundation pruning and fruit pruning also. So this practice can also be followed where the temperature issue is there. So next is that the hail and unseasonal rains, as I mentioned that. So if we see the situation of factors, particularly with your water, so rain and unseasonal rains, hail and unseasonal rains affecting particular area. Now this year we saw that there is a huge loss of grapes in a Nifad area of the Nashik, some part of the Solapur. So these are the pictures were taken from that area where this kind of the situation were arise. Here we can say that due to the heavy rain, the water gets stagnated in the fields, and it affects your other growth stages of the plant. Similarly, there might be the good flowering, but sudden drying of the flowers, or we can say the necrosis. So all these things happen due to the hell, or we can say the erratic rainfall, unseasonal rainfall also. So IPCC, as I in slide I earlier slide, I told that it has predicted that there might be the more extreme chances that there are more chances to get the extreme weather conditions in a particular subcontinent. Here we can see that due to the hail or unseasonal rains, after the fruit pruning, particularly in different stages of various stages, here we can see that it is almost 10 to 12 mm, 10 more than the 12 berry size, diameter of the berry, which have been affected due to the hail. Complete cracking, or sometimes there is a necrosis and drying, all these things. Not only the berry, but we can see that your canes, huge loss of canes also breaking and breakage of the canes and all this. Here we can see the spectral depiction. Similar thing here, we can see that there are the injuries over the canes and arms also on the cordons also, and it affects the next season's growth because heavy hailstorm if there, so it may affect your further process of the grapevine due to the internal injury. Here we can see that. This is the pictorial depiction, and these pictures were taken from the Nashik, Nifad area of the Nashik. So similarly, frost injury is also there. If the temperature goes down less than 5 or 4 degrees Celsius, so definitely the leaf or vegetative growth of the crop may get affected, and similarly bunches will also affected, complete necrosis, or there is a blackening of the berry. It may happen. Some more disease may be there due to the frost injury. So this data were taken in 2012, 2018, and 2022. Similarly, we can see that here the frost injury affects bunches also. Discoloration of the berries sometimes if less frost is there. If the more frost condition is there, so it may get affected due to these things. Similarly, here we can see that also earlier different phases. Already I shown these slides where, so most of the physiological disorders may happen due to the fluctuation in the temperature. So study has been carried out regarding the use of the rootstocks, particularly in grapes. So at a primary level, still the study is going on. So how, if there is a drought or water logging conditions, so what kind of the rootstocks, or which rootstock showing the what kind of the responses, so that has been studied. Still it is in a pipeline. So we have taken the different rootstocks. Doggery is always a Polson 1103P, that is the Polson 110R, 140 Am I audible now? Any participant? Kindly respond. Hello.
Yes, sir. You are audible.
Okay. Okay. So due to the some electrical fluctuation, we could not continue that. So now I will take. So I was telling that the study has been taken at a primary level in NRC grapes regarding the use of the rootstocks, particularly the drought and waterlogging conditions. So here we have observed that 140 RU freedom followed by dog race shows the better response under the drought condition, particularly in relation to the root area. If the treatment is given and the observations taken at a drought condition which has been applied at a 15 days interval. Similarly, in case of the water condition, SO4 as well as freedom shows a better response. Same thing regarding your membrane stability index as well as drought injury index. It shows that particularly your 140 RU 110R shows better response in waterlogging conditions as well as under drought condition 110R and 140. Similar stock shows the better response. So in case of your total phenolic compounds and other biochemicals, that was also taken into the consideration. And anatomical changes which occurs particularly in a root when the treatments are applied. So that has been also studied. And we came to know that in a drought condition, 140 RU, 110R, SO4 followed by Dogri shows better performance. Similarly, Olson 110R as well as 140 RU shows a better performance under the waterlogging condition. So the use of the plastic cover is now becoming more important, particularly in the areas where the high rain or we can say erratic rainfall or hailstorm is there. Some part of the Nashik and the Satara districts, this practice is being followed. So how the material should be there if we are going to use the plastic cover? So it should be interwoven fabric, should have the good diffusion of the light and better spread of the light should be there. It should be oven laminated, ultraviolet stability should be there. So its cloth weight should be there about 140 to 150 GSM per square meter. It should be anti-sulfur in property so that it can withstand in a different kind of the spray of the sulfur also, as well as anti-drip property should be there. So here we can see the photographs where the grape were cultivated under the plastic cover. So study has been carried out in NRC grape with the different treatments, recommended irrigation practices under the plastic cover. 80% irrigation plus your under plastic cover and your recommended irrigation under the helmet and recommended irrigation under the open condition. So result shows that plastic and helmet shows the better performance of the sprouting as well as your other factors, is particularly your yield and all these things we can see in this table. So treatment first, that is plastic cover with recommendation and plastic cover with 80% irrigation of recommended depth shows that good performance in bunch quality as well as yield. So here we can see that under the plastic cover, there is a better bunch load, better sprouting compared to your open condition. Similarly, here we can see that earlier stage of your sprouting and but we can say that the bunch which are coming out from the canes. So as compared to open condition, maximum bunches we can see that under the plastic cover. So this is the pictorial depiction of that. Other your lip water potential as well as sugar accumulation ratio, it has been also influenced due to the use of the plastic cover. We can see here. So what is the cost if we see? So as we know that wine requires your particular trellis system. If any stakeholder have the white trellis system, so they can just give additional support to white trellis. So the cost will be of trellising
So new structures will be near about 1.5 lakhs. Or if we go for erecting the new support structure for the covering the plastic cover, so it will may go 3 to 4 lakhs. And regarding the plastic cover, it is the supportive angles which will be utilized for giving the support to the support for covering the plastic cover. And actual plastic cover requirements will be near about 5,500 square meter. If having the 3 meter width of the plastic cover, so it may cost almost near about 4 to 4.5 lakhs. So near about total cost may go up to the 5 to 6 lakhs per acre if anybody is going for plastic cover utilization in the vineyards. Here we can see the plastic cover, how it is covered. So there should be the distance between the your vegetative growth and your plastic cover. It may vary according to your altitude, or we can say your land slopes and all these things, but almost it should be more than 2.5 feet, so it will be better, so that your vegetative growth may not get affected due to the increase in temperature of the plastic. And there should be the gap between the two plastic covers in the line, almost 1.5 feet gap should be there, so that there is the proper aeration, and whatever the water will be there on the plastic cover, that it will be come down in the middle line of the vines. Here we can see that if we are going to use the plastic cover, so you may get the good quality of the grapes there. And same thing here, we can see the shade net and moisture stress. So it will be better to use if the particular area which is prone to the hail or erratic rainfall in a fruiting season. So what are the benefits of the plastic covers are there? So there is a low risk of staggering the pruning in the particular August to September if anybody is going to harvest the crop early. So low disease may be there, so possible to protect from the frost. High productivity is there, water saving also there, and your biocontrol agents may performs better. It may give a good. They are giving the good results under the plastic cover. So finally, I can conclude here that if we want to manage the abiotic stresses, so there is need of the varietal development. So still, there are not particular varieties which are mostly resistant for the your abiotic stresses, particularly your hail and other things. But the programs are still going on, so may need such kind of the variety, so it will be give a better results. So modulations in the horticulture practices, particularly shifting of the pruning time, as well as your other practices like use of your rootstocks, particularly drought or water stagnation, which will give a better performance. They can be tolerable. So use of the protected structure like the plastic cover or the hail nets. We have to give the effective irrigation practices like recommended irrigation doses or subsurface irrigation or partial right. So at genetic level, so gene isolation that for a specific abiotic stress tolerance that can be very effective, and use of the plant biological means to control the diseases. If stakeholders use these practices, so that abiotic stress management can be done in a better way. So with this, I will stop here. So anybody have the questions, they may ask, please. Any questions are there?
Hello. Hello. Good morning, sir.
Morning, ma'am.
How are you?
Yes, I'm good. What about you?
Thank you for Thank you for this informative lecture.
Yes, ma'am. Please ask.
So, I just want to ask that by using the plastic covers.
Yeah.
So, what was the effect on the GDD and HTU or PT requirements for the grade one?
Madam, your voice is a little bit breaking.
Hello.
Yes.
Sir, was there any effect on the GDD, PT, or HTU requirement of the grade point by using the plastic covers?
Yes, madam. Little bit effect of the plastic cover is there. So there is As compared to your open condition, your harvesting it may take more time, ripening phase and all these things. Near about 6 to 7 days or up to the 15 days, there is a difference as compared to open condition. But the thing is that you will get the good crop and you will get the good yield. So these things are there.
Sir, what about the anthocyanin accumulation and all?
Anthocyanin accumulation. No, madam,
Yes,
it will not get more affected. It will get a good.
but sir, by using the plastic covers, there will be an increase in temperature, and
Yeah.
it is having a direct correlation with the anthocyanin accumulation.
So it varies with your regional conditions, madam. If the frost condition is there, so it may give a different reasons. But actually, in a lower temperature where the grape is grown, so there will be not more effect on anthocyanin content. So it will give a good result, I think so.
Sir, generally we say that there should be a difference of 10 degrees Celsius in the day and night temperature for a better anthocyanin accumulation and good sugar accumulation also.
Yes, sugar accumulation.
How is it plastic cover tackling?
लगेच आणि
Yes, yes.
किती वाजता
Hello.
Hello,
Yes, yes. You are talking about the sugar accumulation and anthocyanin.
sir. And we generally don't have a good accumulation of sugars in our condition. And whenever we talk about the Maharashtra, we see that at the time of from village on up to the ripening, there is a difference of 10 degrees Celsius in the day and night temperature.
Yes.
That leads to better accumulation of the anthocyanin.
Yes,
So
sir.
if we are using a plastic cover,
Yes.
so does that difference vary somewhere because day and night cover is there?
Yes,
So
yes.
how is that thing scattered? That's why what I want to ask.
Yes. See, madam, if we have to save the crop from the abiotic stress, okay? So we have to adjust with a little bit the parameters like you have asked. So not that much difference is there, particularly in the tropical condition. I'm talking about the tropical condition, particularly in the belt of Nashik and Satara and your Sangli district, okay? So
Yes.
actually there is a temperature is not an issue. Due to the use of the plastic cover, only your vegetative phase it may longer, okay? And due to that, there is sugar accumulation is there, but it takes time, little bit near about 7 days to 15 days more as compared to open condition. Okay.
So, sir, by using the plastic covers, our crop is getting earlier or it's getting late?
It will late near about 7 to 15 days compared to open condition.
Okay, is it?
Yes. We have
So
studied.
generally, okay.
We have the data of 3 years, which we have conducted trials in the ICAR-NRCGrapes.
Okay.
But the point is that it is not required to use the plastic cover everywhere. Okay. You can use the plastic cover where the situation of the hailstorm and high erratic rainfall is there, high rainfall is there. In that condition, we can use the plastic cover.
Sir, you are using the plastic cover at a particular stage of a crop or right from the blooming?
Yes, madam. It is depend on your object. If you know that the rainfall is there, because nowadays there is the early prediction of the rainfall is there. Okay.
Okay.
Meteorological department gives the message or the alerts that in the particular time there might be the hail or more rainfall. So farmer use this practice just to escape the rainfall by using the low quality plastic material also. Just they escape the rainfall, just they avoid the injury. So for that purpose, it can be used as a temporary, right from your particularly after your pruning as well as your foundation. When the fruit bud formation is there, at that time we can use. Otherwise, after at the time of maturity, means we can say the horizon period. If we are using the plastic cover up to the harvesting, then it is better. If even though there is a hail or more rainfall, so your bunches will get saved and there will be no cracking of these things.
Okay.
Yes.
Thank you, sir.
So I think one participant have the asked question: if the grape is a temperate region crop, then how the cold injury occurs? Yes. So I already mentioned.
Hello, Dr. Very good lecture.
Yes, sir. Yes, sir. Thank you, sir. So I will reply to.
Very good, very good.
I will reply to this question is that it gives a more better performance in subtropical as well as tropical conditions. It is depend on the varieties also. And as I told you that growing degrades, change in the climate. So definitely there is a change in the crop physiology of grape also. So it is giving the more response in a little bit tropical conditions. So any more question, please? Can you shed some light on the partial root zone drying? Yes, partial root zone drying, it is also one of the irrigation method where as compared to your recommended dose, the little bit irrigation is reduced so that we can save the water. It is simply. But subsurface irrigation will give the more better performance compared to root zone drying irrigation. Any question, please? So should I stop now here? Because almost 10:32 is there. So next lecture will start. Dr. Satisha sir is joined. Sir? We will wait for 1 or 2 minutes. I'll just contact to the next resource person. So next lecture is that management of the physiological disorders of grape. So the lecture will be delivered by Dr. Satisha sir. He is a principal scientist in IHR Bangalore. So he will join soon within 2 or 3 minutes. So just we have to wait for him. one participant, Soman, I think she or he is asking the question about is the breeding approach lecture over? No, no. It is just shifted. So it will be there tomorrow in afternoon session. Dr. Gawande, scientist, breeder, plant breeder, he will take this lecture.
Hello.
Yes, sir.
I'm ready.
Yes,
Sorry.
yes, sir. Okay, okay.
Sorry.
So, dear participant, now Dr. Satisha sir is now connected here. He's with us. Sir, welcome to you in this training program, sir.
Thank you.
So, sir, he is doing work on grape only. He was earlier in NRC grape. So he has done a lot of work on the basically on the rootstocks, canopy management aspects, enzymatic profiling, and all these things. So, sir, I will request you, sir, kindly deliver the lecture on now. The topic is that physiological disorder management in the grapes.
Okay. Thank you, Dr.
Yes, sir. Yes.
Nishant.
Prashant.
Prashant, sorry.
Okay.
You are two. Nishant is also there,
Yes,
no?
sir. Yes, sir. He's
Okay.
Please,
So
sir.
I will share.
Okay.
Is it visible?
Yes, sir.
Okay.
It is opening, sir.
Yeah, it's opening now.
Yeah, it is.
Okay. Good morning, all. So may I know who are all the audience here?
Sir, most of the participants are students, MSc, PhD,
Okay.
some assistant professors from SAUs,
Okay.
and few agriculture officers from the state agriculture department, sir.
Okay.
Yes, sir.
Okay. Thank you. So we will start. Good morning, all.
Sir,
Today,
अभी तक ओपन नहीं हुआ आपका?
open नहीं हुआ?
Yes, please try again. मतलब आपका जस्ट वो दिख रहा है, but it is not opening. अभी हो रहा है कुछ.
हो रहा है क्या? मुझे दिख रहा है इधर.
सर यहाँ पे नहीं दिख रहा है. Any participants? Response from any participants?
No, sir, screen is not visible.
Not visible.
It's
Sir,
not visible. Stop.
Sir,
Again, I will start.
Again करिए sir.
Now?
Yes, sir. Sir,
Yes, sir, it is visible.
presentation mode में लाइए, slide mode.
Is it?
Yes, sir, now it's visible.
Now it's visible?
Yes, sir, it is visible, but वो slide mode में नहीं है, sir.
नहीं, मैं slide mode में डाला हूँ ना. अभी देखो. For me, it is slide mode.
Arusha ma'am?
Yes,
it in slide
sir.
mode? Is it in slide mode?
No, sir, it's not.
Yes, sir. हमें तो नहीं.
But I made it slide mode only.
Okay, sir, but it is visible now.
Yeah. Then shall we start?
Yes, sir. Yes, sir.
Okay. Because for me, it is in slide mode only I am presenting.
Okay, sir. Okay.
Okay. So, good morning again, once again. So, today we will discuss about some of the important physiological disorders in grapes. Before that, we should know what is physiological disorder, because many of the farmers sometimes they will bring some samples, leaf samples. It is look like yellowing. It may be they will think that it is a downy mildew or nutrient disorder or something like that. When we analyze everything will be positive, and negative means there won't be any disease symptom, there won't be any deficiency or excess of the nutrient, everything will be normal. Still, there will be some symptoms. So those kind of symptoms we may be attributed to some of the other factors other than the biotic and abiotic stresses. So in most of the fruit crops, yield reduction or quality deterioration of the fruit crops mainly because of biotic and abiotic. You know, in grapes, yield reduction or the quality reduction is mainly due to either drought or salinity stress or some of the pests or diseases. And sometimes even pre and post harvest disorders also reduces the marketable yield and quality. But sometimes other than these factors like abiotic stresses or pathogens or pests, there will be some factors beyond some optimum range that may leads to deterioration of physiological process. This kind of the variation from the normal, that is the variation from the optimum range, causes some of the substantial changes in the plant physiology. They are referred as a physiological disorders. that is not due to either pests or diseases or excess or sufficient nutrients or something. It is somewhat which is normal, not abnormal growth pattern of external or internal conditions of fruits caused by adverse environmental conditions during either growth of the crop or during the ripening or during the harvest, storage or marketing.
Sir, just
Ah.
interrupt you, sir. Sir, your slide is not moving.
No. I'm explaining.
Okay.
I'm changing.
Okay.
This is just introduction I'm giving. So those symptoms of the disorders, these physiological disorders may appear like disease. That's what I was telling. Some of the disorders symptoms, which it may look like disease also, or nutrient deficiency or excess, but when we analyze or when we see the pathogens, there won't be any pathogens, or either nutrients will be deficient or excess. Still, they show symptoms. They are because of the some of the variations caused by environmental conditions or some other factors. So they are called physiological disorders. And usually physiological disorders are often caused by either deficiency or excess of something that affects metabolism, something which presence of something, that something we have to identify. That's why it is always question mark whether it is a physiological disorder. First, we should see whether it is a pathological problem, entomological problem, or nutrient deficiency. If everything is normal, then we can relate to some other factors, and it can this physiological defect disorders can affect at all stages of the vine development. And unlike pests and diseases, these physiological disorders cannot be transmitted. Means from one plant to another plant, these physiological disorders cannot be transmissible. And also the physiological disorders can occur because of the some faulty practices or the treatments which must have been given in the past season or previous year. That consequence can appear during this year, and generally these physiological disorders, if in a bunch, if in a leaf or some in a plant, if one only portion of the vine or a bunch may be affected, and a normal bunch will have a exact proper color, sugar, quality parameters, everything. But that means it cannot transmit from one place to another place. And sometimes these physiological disorders can act as an open door for pathogens. That means they can be a predisposing factors for actual physiological disorder. So this you have to keep in mind. Some of the there are different types of physiological disorders based on some may be due to variation in the atmospheric conditions, and some may be due to edaphic means what? Soil factors. So based on the causal factors, we can classify the physiological disorders. On the basis of causal factors, it may be due to temperature related, like injuries of fruits resulting from the extreme temperatures outside the optimum range. It may be higher temperature or low temperature, and it may be occur either within the vineyard or after harvest in the post-harvest period. So these various disorders may be, sometimes it may be considered as a freezing injury, chilling injury, heat injury. These are I am telling different types of physiological disorders that apply mainly due to the variation in the temperature. And any physiological factors like temperature, nutrients, if present beyond the required range, they will disturb the internal normal metabolic process of the plant, will be disturbed if there is any excess temperature or excess nutrient, it may happen. And sometimes it may be physiological disorder usually occurs in two phases. One is induction phase, means just it will trigger, and another is developmental phase, means sometimes it will be hidden. When the environmental condition is favorable, it will show its symptoms. So it is a induction phase is just triggering, and after that it will show the development. So this is the one type of physiological disorder which happens because of the variation in the temperature. Next is nutrient related disorders. Common in most of the fruit crops is there is a deficiency of calcium.
Sir,
Is it?
are you moving the slides?
Yeah.
Sir, it is not moving. It is still showing
See
the
here.
first slide.
Now it is nutrient related disorder has come.
No, sir. First slide is only title.
I'm moving. It is visible to me.
It is not visible, sir.
Ah.
You just share it again.
Now,
Yeah, now moving, sir.
if it is in what is happening? No, if this is visible, I can present like this, because when I apply in when I go to slide mode, no, I think you people are not. Here it is showing.
Okay, okay, okay.
So is it visible to all? Means I will.
Yes, sir. Now your slide, third slide, sir. Physiological
Yeah.
disorder, often
Yeah.
caused by. Yes, okay. Move, sir. 1-2 slides again. Yes, yes. Type of physiological disorder. Okay, moving.
So here, if I go to slide mode, it is not moving to you people.
No, sir. This is okay, sir. Now
This is okay means then I will continue like this.
Yes, yes.
Okay. So I was mentioning about nutrient related disorders. It may be due to imbalance supply of essential nutrients, like and in most of the physiological disorders, like in many fruit crops, calcium and boron play major role. All of you know calcium is very much important for cellular integrity. So deficiency in calcium causes some of the disorders. One classical example is bitter pit in apple. Similarly, in our grapes, it is deficiency of calcium and boron. Calcium causes what? There is a harvest, pre-harvest berry drop will be there, and boron deficiency causes short berries or water berries. These are the major disorders. And zinc is also one of the important micronutrient which causes some physiological disorders, because all of you know zinc is responsible for synthesis of oxidants like indioacetic acid, that is IAA. And the next category of disorders is atmospheric related disorders, like variation in the macro and microclimate within the canopy. You know, in microclimate means what? What is the temperature, relative humidity and light within the canopy is called microclimate. Sometimes there may be variation in this climate. One classical example in grapes, where due to variation in macro and microclimate, is our pinkberry. That is Thompson seedless and some of the its mutants like Tasha Ganesh and Sonata, they show pink berries in the green bunches. So that is not a disease, that is a pink berry disorder. That is because of the extreme variation in the temperature. Sometimes even very high or low concentration of carbon dioxide also causes some of the physiological injuries. It will interfere the normal metabolism of the plant, and that may cause some of the variations in the normal growth and development of your berries. And many fruits are highly sensitive to ethylene and get deteriorated upon ethylene exposure. So there will be some ethylene toxicity in some of the fruit crops, and some of the presence of some of the chemicals like ammonia, sulfur dioxide, methyl bromide, they also causes some of the physiological disorders. And these disorders are commonly called as CO2 injury, ethylene injury, ammonia injury. But in our grapes, since it is a non-climateric fruit, means a carbon dioxide peak or respiration peak or ethylene peak is not there. So we don't get most of the ethylene injury in our non-climateric fruits. So this is in brief about the different types of physiological disorder. Whenever you have to study the physiological disorder, first you should make sure that it is not due to either diseases or it is not due to major nutrient deficiency or excess. After analysis of that, you have to come to conclusion that it is not a disease, it is a disorder. So some of the important, there are a lot of disorders now and then grape growers are facing, and I will discuss one by one. Major important which will cause huge loss or which will have a drastic effect on the normal growth and development of the grape vines. So such disorders only I have selected for this today's lecture. First one is, so first classification is ecophysiological disorders. In that, common thing is dead arm, and trunk splitting. This is one of the disorder. Second one is barrenness of vines. Means barrenness means vines, there won't be any sprouts. All the, even after normal pruning also, you can see lot of empty cordons in your vineyards. So that is called barrenness of the vines. So next is sun scald or sun scorch or sun scald. This is due to excess heat or sunlight, direct sunlight exposure to bunches and poor cane maturity. Even after 90 days or 100 days also, some of the canes will not mature. They will remain in green. So that is the one of the important disorder which significantly affect the growth, your yield of the bunches. And the next one category is physiological disorders. These, I think many people, those who are well versed in grapes, they know water berries, short berries, pink berries, bud and flower drop, and post-harvest berry drop. These are major physiological disorders. Some of the nutrient disorders are cluster tip wilting and bunch stem necrosis. So these are the some of the important physiological disorders in grape cultivation many of the grape growers are experiencing. Apart from these, there are a lot of other disorders that are caused due to some of the extreme climate conditions like hail damage. Nowadays you are seeing frequently hailstorm will be there. For that also causes significant physiological damage to some of the internal organs in the plants, and they will put forth lot of physiological changes in the crop. So that is one. And also some of the herbicide injuries. When we spray excess herbicides, because of some of the drift also, that herbicide injury you can see in many of the vineyards. That is also one kind of the disorders. And you can see black leaf symptom. Black leaf symptom is also due to the excess potassium application and drought stress. And red leaf is also there. Like that, lot of disorders are there. But commercially important, which will have an economic impact on the grape berries, grape production, I am discussing. Let us see one by one what are all the important physiological disorders. Hope everybody are understanding whether all of you are following.
Yes, sir.
Hello.
Yes, sir. Yes, sir.
Is it okay now?
Yes, yes. It's okay.
Okay. Then I will go one by one now. First physiological disorder, very important in is dead arm and trunk splitting. Means what happens here? Whenever there is a severe moisture stress. Nowadays you are seeing, I think entire India is facing severe drought. So I don't think in Maharashtra also it is drought condition may be existing with high temperature and all. So what happens now after harvest? If a severe moisture stress is experienced in the vineyard, and because of high light and temperature intensity, these are the predisposing factors. If such environmental condition exists, immediately after back pruning, what happens? Usually, your bunches will, vines will start sprouting within 15 days to 20 days. Your bud burst will start. But when the predisposing factors like high temperature and light intensity is there in your vineyard, there will be, even after 3 to 4 weeks of pruning, the sprouting will not be uniform. And even if they sprout, after 3 to 4 weeks, there will be slowly drying of the leaves. The entire shoot will dry. If excess drought condition prevails, there will be cardan splitting. You can see here some of the cardan start splitting, and even the trunk, dead arm and trunks will start splitting. You can see in the lower picture, you can see some of the vines on the cardans, there is no sprouting. This is the this one, dead arm we call it as. So what happens here, the translocation of the water and mineral from the trunk to the arms will be stopped because of the continuous stress and also high temperature and light intensity. So this is associated with high sunlight intensity, temperature, and low atmospheric humidity. So, but many people thought that this may be a fungal diseases also. So they have analyzed some of the fungus, whether any fungus is causing this dead arm and trunk splitting, but no causal or no specific fungus has been identified. So then they have attributed it as a physiological disorder. So this will ultimately results in reduction of the yield, because once sprouting is not there, or even the death of the shoots happens, no further growth will be there, and it will have a physiological there will be drastic reduction in the yield due to this disorder. So how we can overcome this disorder is, control measures means suppose if there is a, because you in most of Maharashtra and all, we know a lot of the climatic predictions are there. If so, one of the means to reduce this trunk splitting disorder is early summer pruning, so that before the temperature rises beyond 40°, your sprouting should happen and the leaves should be almost reached 5 to 6 leaves or 7 to 8 leaves. Then we can, once they have self-sustained, it is they can overcome this splitting of the arms. So early summer pruning is one of the control measure. And in extreme high temperature, whitewashing with our normal lime, agriculture lime, whitewashing of trunk and cordons can reduce the intensity of sunlight because by reflecting the direct sunlight. So whitewashing you can do on the trunks and cordons in severe summer conditions, and maintaining adequate, if irrigation is there, try to give irrigation, because what happens after harvest, most of the vineyards will be get neglected. So through periodic irrigation during summer pruning, this is very critical. Even if your borewell is, somehow you should manage good soil moisture during the, at least during the back pruning period, so that sprouting should happen normally. Then we can reduce. And during the back pruning, it is the time when you can correct some wrong train procedures, like removal of the dried arms, cordons, everything you can remove, and you can start new cordons. So this is the time you can adjust the pruning, so that we can remove all the dried arms and new cordons can be established. Or else what happens, every year this dead arm keep on accumulating on the vines and result in the splitting and drastic reduction in the yield. And adjusting the position of the canes, because what happens if the since if it is a very horizontal canopy, direct sunlight will be falling on the shoots, and they will more prone for the sunlight. So it should, we should position the canes to the vertical, white trellis or V trellis, so that the wider angle exposure them to direct sunlight will be there. So you try to position the shoots so that direct sunlight won't come and fall on your cordons. So this is some of the control measures we can follow to reduce the formation of the trunk splitting and dead arm. Second one of the diseases, disorder is barrenness of the vines. Barrenness means what? There won't be any shoots on the vines. See here, these arms are the cordons. Here and there you can see some of the plants which are barren. Barren means was nothing will be there. So this is usually occurs in some older vines after 15 to 12 to 15 years of the this one by establishment of orchard. Some of the vines start becoming barren, where cordons will be barren and without sprouting of the buds. These are the typical barren vines. So in entire vine, we could observe too much of the barren wood and inadequate number of shoots. You can see, if suppose by 10 by 4, 10 by 6 shoot canes, vines should have minimum 40 to 50 canes. But hardly you can see any canes. Sometimes you can see only 10 to 15 canes. And most of the canes which are produced will be unfruitful, means they are not fruitful canes. And after fruit pruning, we could observe production of only unproductive canes or vegetative shoots. This barrenness of vines, we should attribute this to wrong viticultural practices. That's what I was mentioning in my previous disorder, that is the trunk splitting and dead arm. Proper training and pruning is very important for entire lifespan of the vine. So wrong viticultural practices, like wrong pruning practices. So during back pruning, we have to rectify. We should remove dead arms, disease arms, weak arms, everything we should remove. If you keep on continuing those without rectifying the problematic canes, then in long run, this will pose a problem, this barrenness. So barrenness in vines is mainly due to failure of buds, bud failure, and also due to poor fruit bud differentiation after back pruning. So this is the barrenness of the vine. So what are the important causal reasons for barrenness? As I mentioned, defective training and pruning systems, that is faulty pruning practices, and inadequate care. I'll give 1 or 2 examples for this inadequate care during, in most of the northern Karnataka and your entire Maharashtra, we will take 2 prunings, that is back pruning and forward pruning. Back pruning, why we will do? To maintain, to establish good fruitful canes. But since we don't get crop during that after back pruning, there may be little negligence of the cultivation practices like judicious application of fertilizers, nutrient management, water management, and growth regulators. If there is a negligence, that is reflects in bearing period. Means because why in after back pruning, what we will do? We will give lot of nutrients and also growth regulators like CCC, uracil. They all should be given for better fruit differentiation. If you fail to practice such good practice management practices during back pruning, after forward pruning, you can see only barren canes, means non-fruitful canes. And sometimes another reason for barrenness is bud failure, means failure of flower bud formation, flower bud killing, and failure of sprouting of flower buds. So these are some of the reasons for barrenness. So how we can overcome the barren means proper pruning and training practices, shoot positioning, and corrective measures during back pruning. That's what I was telling, like removal of barren cordons and rectifying the, means we have to correct everything. Faulty canopy should be rectified by encouraging new cordon formation. No problem if we lose some of the canes during back pruning, but in long run, to sustain the productivity of grapes, we should rectify the some of the non good management practices which are not followed. We can be rectified during back pruning. Why we do back pruning is one of the objective of back pruning is also to correct the wrong management practices. Whenever there is a non-productive cordons, disease cordons, or training is not properly done, that time you can correct everything during back pruning. And proper management practices for fruit bud differentiation stage. As I was telling, during the fruit bud differentiation stage, that is 40 to 60 days after back pruning, you should follow the subcane development, spraying of CCC, that is to reduce the vigor and also to improve the fruitfulness, and spraying of 50 ppm uracil, and good application of phosphorus, and some judicious water management practices can improve fruitful canes after forward pruning. And one of the reasons for barrenness is overcropping. Suppose in 1 year, if you take more crop, definitely during second year, plant vines will get exhausted and they won't get proper nourishment. And next year, the vines may be barren, means non-productive canes may be more. So we should do the good crop regulation practices like bunch thinning, berry thinning, and maintenance of proper. Here comes the source to sink ratio. So we have to have a sufficient number of canes, and number of leaves on cane should be optimum for producing a good quality fruit. If the source is less and sink is, means what happens? Plants will get lot of exertion. Most of the bunches will exert all the nutrients, and next year vines will become barren. So this is the barrenness of the vines. Third, one important disorder is sun scorching or sun scald. You can see here the bunches, they are directly exposed to hot sun. They will change their color. Normal green berries, they will start doing turning into amber color. Then this is more severe in red globe grapes where canopy is very less or meager. Here sufficient leaf area won't be there. So even in some of the wine grapes also, we can see lot of the sun scorch. It is what it will do is it will discolor the grape cluster. It may be in colored varieties, it may be very brown or drying, but in green varieties like Thompson seedless, they may start browning, and after that there will be cracking and shriveling of the berries. Although sun scald is abiotic stress, sometimes, because, as I told in my beginning itself, most of these physiological disorders are predisposing factors. They will act as an entry point for infection of some of the pathogens. They will make some entry points for infestation of some of the pests and diseases. So whenever there is a physiological disorder, it will be followed by some of the diseases like rotting may be there, some cracking may be there. So all these are because of the sun-scald damage, and it can be a significant problem in very areas where the intense sunlights are there, or where there is a heat spikes in cooler regions. Sometimes what happens very abnormally in cooler period also there may be high sunlight intensity. So direct sunlight on the unprotected clusters, they began to develop these symptoms of browning, and sometimes drought conditions also increases this effect, as well as management practices. There is in wine grapes, to improve the quality, what they will do, sometimes they will do some leaf removal practices and also shoot thinning practices. If they do excess, they may directly expose to sunlight, and they may result in and wrong row orientation. Row orientation also plays major role in formation of the sun injury or sun scorching. So how we can, so you can see if this in sun scorching, if exposed bunches and shaded bunches, when we analyze the nutrient contents or chemical, its quality parameters, though all the parameters are very good, but their shelf life will be less, because the exposed bunches will have greater TSS, anthocyanin, phenols, and flavonoids, whereas they have lower in titratable acidity, malate, juice pH, berry weight, potassium. Whereas in shaded bunches, your TSS will be less. But these exposed bunches, if it is very high, also what happens? Its shelf life will be very less because of the high temperature. So this is the difference between exposed and shaded bunches. So how we can control the sun scorching means, first and foremost is, most of the vineyards we should establish row orientation should be north to south. If we orient the rows in north-south direction, so we can avoid the direct exposure of the western sunlight, because in grapes, western sunlight is more injurious and potential hazard than eastern sunlight. So north-south orientation can reduce the incidence of sun scorching to some extent. And now in lower portion, you can see they have some shade nets. If we spread or install some shade nets, we can reduce the cut off the intensity of light which is falling directly into the shoots, and sometimes shoot positioning of the shoots to cover the bearing shoots, because in most of the vines there will be both bearing and non-bearing shoots. Some bunches canes will have bunches, and some don't have bunches. Such canes which do not have bunches, that can be positioned to protect the exposed bunches. And sometimes we can use in heavy severe scorching period, if we use some sprinkler and micro sprinklers, we can reduce the temperature by 5 degrees Celsius, Fahrenheit or Celsius. So, and sometimes spray of particle film forming, some of the film like anti-transparent chemicals like Calvin, they form thin coating on the clusters, and they can reduce the intensity of sunlight, and that they prevent the sun scorching. And to protect the bunches which are exposed directly, we can cover bunches with some of the paper covers. Earlier they used to use newspapers. Now because it is coming under this scrutiny of food safety, covering the newspaper is not a good practice, but there are some brown paper bags which mostly used in covering for export grapes. Such covers, paper bags or butter paper bags or brown bags can be used only for bunches which are directly exposed to sunlight, so that the sun scorching can be reduced. So this is about sun scorching. And one more ecophysiological disorder which I was mentioning is poor cane maturity, because for wines to get good carbohydrate, your cane should be fully matured. Only matured canes can give good sprouting, and also fruit bud differentiation will be good in the matured canes rather than the young immature canes. In this type of disorder, shoots fail to mature. You can see here, even when the berries are formed and the berries are almost developed, but still canes have not matured. They will stop maturing at the off period only. So And such shoots also start turning pink and red due to low temperature in winter also. It is more serious in vineyards where most of the shoots, more number of shoots are there. The very dense shoots means the shoot density is very high, and also closely planted vines, and excess nitrogen and irrigation also causes this poor cane maturity. And this one of the other disadvantages of this poor cane maturity is cane maturity is not there. They are mostly green in color, and they are mostly susceptible for some of the diseases like powdery mildew, which ultimately results in dieback of the means there will be death of the canes. So poor cane maturity indirectly affects your berry quality because there will be reduction in the TSS, anthocyanin, phenolic compounds, and nutrients, everything. Those bunches will be very insipid and watery. So shoot immaturity was found to often associated with premature leaf fall, means before maturity of the leaves, leaf fall will be there, and excessive number of shoot growth will be there. Wherever there is a dense canopy, there are more, and the canes which are in the deep side of the shade, they will have this problem of poor cane maturity. So how we can overcome this poor cane maturity is, in order to avoid cane maturity due to overcrowding, shoot thinning. Because I think from yesterday you are listening about most of the practices, canopy management practices. One first and foremost important practice in back pruning, after back pruning, is shoot thinning. If you do the shoot thinning at right stage, immediately after pruning, before 7-8 leaf stage, before that, if we thin out the excess shoots, then remaining shoots will start producing normal growth, and they will mature and attain maturity at 90 days. If you don't do the shoot thinning, so we have to maintain optimum shoots per unit area. So in the wine canopy, I think in canopy management class, some of the scientists must have explained already what should be the shoot density per unit area. And vines may be trained to vertical or open trellis rather than horizontal trellis. And more number of shoots can be retained per unit area of the shoot. Hello? Somebody stop speaking, please.
Hello, madam. Please mute yourself, ma'am.
Okay.
Please, sir.
Okay. So shoot thinning is very important to reduce this poor cane maturity, and excess nitrogen and irrigation fertilizer should not be applied at 40 to 70 days, whenever there is a fruit differentiation stage is there, that is the right time for turning that green color into the brown color. During that time, please reduce the concentration, your quantity of nitrogen fertilizers, and also give mild stress for those canes, because mild stress and low nitrogen fertilizer is also required for your fruit bud differentiation and cane maturity. Instead, after 70 to 80 days, you start your potassium fertilizers. So by following this best management practice for poor cane maturity is avoiding excess shading and excess shoots. So shoot thinning is the one of the prime important practice you should follow to reduce the avoid the poor cane maturity. So these are all about ecophysiological disorders. If you come to the berry phase, we have several disorders. One important disorder is waterberries. This waterberry, it can be observed during the ripening period, because it will look like normal. The growth and development of this berry bunch will be normal, but what happens? These berries will not be very attractive. They will be very pale in color, and it is observed in berries are almost normal in size like other bunches, but the flesh, if you press it, it is very soft. This water berries will be very soft compared to your normal berries with watery texture, and they are not attractive color. They are very dull in color, and by the time of harvest, they will be start wilting, withering and wilting. There will be shriveling of see here at the lower this water berries start wilting, shriveling, and they will dry. But and what are the major predisposing factors? Means some of the factors which are responsible for formation of water berries are excess shading. These water berries are seen in the bunches which are developed at the excess shading portion, canopies, and also too much moisture and humidity during veraison period. Whenever there is a too much moisture, water stagnation is there, there is high relative humidity during veraison period, it will cause water berries. And excessive irrigation and nitrogen fertilizers during berry development, because we will give more nitrogen only till 40 to 50 days after pruning. After that, we should drastically reduce the nitrogen dose, and we should increase potassium, phosphorus followed by potassium. So, and also same is the case with irrigation also. So at the time of grand growth period of the berry, if there is excess nitrogen, what happens? There will be lot of formation of water berries. And one of the other reason is insufficient source to sink ratio. Means if the bunches are more and leaves are less, there will be starvation of the berries. So there won't be good formation of enough sugars. So there will be reduction in the this flesh or pulp formation. The accumulation of the lot of water will take place and results in water berry formation. So control measures is what? Proper application of potassium in right quantity at right time, which can reduce the water berry incidence. And crop thinning, because in grapes mostly you will get excess bunches only. So you should be very lenient and thin out the excess bunches depending on the requirement. If they in a single shoot, if there are 3 bunches or 2 bunches, depending on the leaf area availability and also cane thickness, please thin out the excess bunches. If you don't do the excess bunches, this will have a water berry formation, sugar accumulation will be problem, and it also causes reduction in the quality of the bunches also. And shoot thinning to reduce the buildup of excess shade in the bunch zone. That's what I was telling. Most of these physiological disorders can be overcome by good management practices, right from the planting till pruning, training, and also shoot thinning. We can measure, follow the right training and pruning practices and correct application of nutrients at right time in right quantity can overcome most of these physiological disorders. So this is about water berry. And this is, and one of the other important disorder is short berries, and it is also commonly called as hen and chicken disorder. Why we call hen and chicken disorder? You see here in this cluster, we can see some big berries followed by small clusters. The small berries are there. So this big berry, we call it as a chicken, sorry, hen, and the surrounded by small berries, we call it as a chicken. So this is also called as hen and chicken disorder or short berries. Usually in normal bunches, some of the berries are extremely small, soft and seedless compared to other berries. So what are the attributing factors for this short berries is boron deficiency. Though boron is required in very minute quantity for grape, but still it is a crucial for boron to be there, especially during pollination and the fruit set stage. If there is a deficiency in the boron, deficiency in the boron or incorrect, and so there will be formation of the poor pollination and pollen growth, because boron is required for pollen growth. So if there is no proper pollination, what happens? Some of these symptoms will be appearing. And also GA3 application, wrong application of GA3 at the wrong time. Because you know schedule, I think you must have already learned about schedule of GA3 application. If the GA3 is applied at wrong concentration and at wrong time, that also leads to the formation of short berries, and also sometimes poor pollination and fertilization, some defective flowers, and improper carbohydrate nitrogen ratio. So I told you should maintain proper leaf area. Thumb rule is that one shoot should have minimum of 10 to 12 leaves. Proper good leaves can nourish good bunch. So poor pollination, reduction in the carbohydrate content and increase in the nitrogen can cause short berries. And usually short berries are seedless and very small in size. And this disorder, as I mentioned, this disorder is also known as hen and chicken disorder. Because why? Because of the boron deficiency and also zinc deficiency. So how we can overcome this short berry formation is application of, because I told proper pollination and fertilization is very important. for fruit set, better fruit set and normal development of the grape berries. If there is a reduction in the pollination and fertilization, because of that, fruit set will be less. So application of some of the auxin compounds during pollination, wherever the some of the varieties are known to set these short berries, in such varieties, if you apply auxin, we can reduce the formation of short berries, and application of GA3 at right concentration and girdling, because I told carbohydrate starvation also causes short berries. So sometimes we use, now girdling is not that much practiced, but whenever there is a severe short berry formation, in next season you can take some girdling measures to retain the carbohydrate to the bunches, and application of micronutrient mixtures like zinc and boron, they can reduce the incidence of this short berry. And one more important disorder is flower and bud drop. It is because this was first identified by in French France. It is called as shelling or color, COULURE. This is the flower and bud drop. Usually in grapes, what happens immediately after full bloom, you can see if we do some shaking of the clusters, there will be normal drop of the berries, flowers and buds. So that is normal. If the 10 to 15% of the shedding is there, that is normal. If there is a excess, the more than 15 to 20 degree, so 15 to 20% of the bunches start, flower buds start falling, that is a serious physiological disorder. And it is mostly very common in some of the northern region specific varieties like perlite, beauty seedless, but not much common in our South Indian varieties. And what happens after expansion of the panicles, the flower buds starts before pollination, and immediately after fruit set, they will start falling. So you can see in the lower picture, you can see it will only rachis and rachulae will be there. Entire flowers and flower bud will drop. Can you see? No. So this is the flower, severe flower and fruit bud. You can see here in the lower picture, and only rachis is there without any fruit or flower. So this is the typical flower color type of the disorder. So the some of the predisposing factors which are responsible for this is low level of nitrogen. Usually nitrogen, because I told in the beginning we should give more nitrogen, and insufficient growth hormones like gibberellins and auxins and cytokinins, along with some of the environmental stresses, they can cause this flower and bud drop during the early season. And control measures is, before full bloom, we should not stress the vines for long period of time, because it causes abscission layer in the flower clusters and they may fall. Similarly, excess moisture in the soil along with salty irrigation also causes flower drop. So judicious water management is one of the important practice to be followed to reduce the flower drop, and foliar application of some of the micronutrients like zinc can be given, because I told auxin, if there is a reduction in the auxin, there will be formation of the abscission layer. So if we spray the zinc indirectly helps in synthesis of tryptophan, which is the precursor for auxin synthesis. So when auxin level increases, abscission level will be, means that leaves flower bud senescence will be reduced, so they can retain, or else what happens, they will fall. So this application of zinc can reduce this, and exogenous applications of NAA and IAA, sorry, this is not IBA, it is IAA, before flowering, as can reduce the abscission in Thomson seedless, and usually higher nitrogen level by reducing CN ratio in the shoots, because here it is not the either carbohydrate or nitrogen. The CN ratio should be proper. So application of high nitrogen dose should be avoided, and excessive soil moisture and more water stress, both extreme conditions like Because I told ethylene or abscission formation is usually formed due to stress. If we give flooding or excess soil moisture also induces ethylene, even stress also induces ethylene. So it should be optimum. Water moisture, soil moisture should be optimum so that abscission layer formation will be reduced. So this is the control measure for flower bud and flower drop. Next important is pinkberry, and this is mostly restricted to your Thompson seedless grapes and its clones in mostly Maharashtra and northern Karnataka regions. What happens here? The berries in entire berries, only few in entire bunches, only few berries here and there. First, there will be formation of the slight tinge of the pink color. Pink color will start, then it will spread slowly to the entire berry, and but the other surrounding berries it won't be pink. Here and there, very sporadically in entire cluster, there will be formation of the pink berries, and it is usually start forming at the veraison stage. When the berry starts softening, there will be formation of the pink berries. And the one of the main reason attributed for pink berry formation is whenever there is a diurnal variation, very too much variation in the day and night temperature. That's what diurnal variation means. What? Night, if the day temperature is beyond 32, 34 and night temperature, the difference between day and night temperature should not exceed 15 degrees Celsius. Usually during February, what happens? Sometimes day temperature is also very high, but night temperature will be very cold. So the variation will be more than 15 degrees Celsius. During that time, one of the theory for pinkberry formation is that too much diurnal variation in day and night temperature also is one of the main cause for pinkberry. But the research is going on from past 50 years, but the exact reason for pinkberry formation is not known. We have to do the management practices. These are all the assumptions. And direct exposure of sun, sometimes they used to tell these pink berries are formed only in the exposed bunches. But when we went for survey long back in 25 years back, I think in NRCGrapes there was one huge project was going on along with MRDPS scientists people. We did lot of survey. There is no clear-cut formation of pinkberry. It is restricted to external bunches. Even in the bunches which is placed in the deep shade also, there was a formation of pinkberry. So what it causes is, though pinkberries are very tasty, but their shelf life is very less, and it mainly affects export grapes because export grapes requires very clear uniform color green or amber colored bunches of, but here it will be a very unacceptable. Market is unacceptable and it will fetch lower market and shelf life is very less. So these are some of the theories. In those days, Dr. Kilari was working a lot on pinkberries. According to him, he considered as ecophysiological disorder. It may be due to nutrient and hormone imbalance, ethylene mediated phenomena, excess use of hormones, high sucrose, high mineral status. So these are all some of the thoughts that they studied, but still there is no clear-cut understanding about what is the formation of pinkberry. Finally, this control measure, how we can control is, though we don't know the exact cause or reason for pinkberry, we can do some recommendation. Like I think in NRCGrapes also and many farmers also, they are practicing this. They will cover the bunches immediately at the time of ripening. After ripening, tie-wet bags and non-fabric own fabric bags will be, they will cover the bunches with these bags so that they will avoid the direct high temperature also, direct sunlight also, so that and one of the management practices what we can do is adjusting the pruning date. It should not coincide with the extreme diurnal variation. I told no that temperature difference between day and night temperature, it should not be beyond 15 degrees Celsius. So accordingly, if we adjust your pruning practice also, to some extent we can reduce the extent of pinkberry, avoid soil moisture stress from berry softening. During the berry softening, that is veraison stage, try to avoid moisture stress and give adequate dose of potash during 30 days prior to, that's what I was telling, from 70-80 days itself start giving potassium fertilizers. And one of the other recommendation is spray of BA, dipping of the bunches with benzyl adenine at 10 ppm at berry softening stage. As you know, benzyl adenine is a cytokinin compound. Cytokinin compounds will help in retention of the chlorophyll. Means they will reduce the formation of ethylene. They are antagonistic. Cytokinin and ethylene are antagonistic. So if we give the benzyl adenine, they can retain the chlorophyll in the berry skin, thereby reduce the formation of pink berry by reducing the loss of chlorophyll. So this is the control measures for berry pink berry formation. And another important physiological disorder is post-harvest berry drop. So this problem usually occurs in some of the varieties like Manjari Navin. I think the NRC grapes also will accept this. The people are Dr. Deshmukh, you can this Manjari Navin is susceptible for, because after harvest, when we harvest and keep it in the tray, there will be drop of the berries. In Thompson seedless also sometimes we can see, observe these Sharad seedless. These are the susceptible varieties. What happens? This is attributed to poor pedicel attachment. Pedicel is what? That fruit stalk. That attachment to the rachis is very loose, so that just if we shake the bunches, there will be dropping of the berry. And it will also, it is the major problem during the storage of the grapes. And the causal or exact cause is endogenous hormones like higher accumulation of ABA and ethylene may aggravate this problem. And also the wines which have a calcium, less calcium content, because you know, calcium is the one which has a good cell wall component, and pedicel attachment can be increased with the application of calcium. So this is the main reason for post-harvest berry drop. So control measures is spray of naphthalene acetic acid, that is Planofix we call, 50 to 100 ppm 8 to 10 days prior to harvest can reduce the post-harvest berry drop. And right application, that's GA3 at 3 to 4 mm and 8 to 10 mm, because GA3 also helps in increasing the cell wall elongation and the pedicel attachment. Similarly, spray of calcium nitrate, as I told, calcium is the one which is required for berry attachment. If we spray calcium nitrate at 0.5 to 1% at veraison stage, we can increase the cell wall integrity and pedicel attachment, thereby reduce the berry drop. And another important is cluster tip wilting. You can see in most of your Thompson seedless and in some of the wine grapes, this is common. Only this tip region of the bunches, they start wilting. They will dry, they will turn brown in color, and they will wilt. So at the apical end of the rachis, we can see there won't be any conductivity of the water and nutrients to this apical portion, and they will start wilting. So in severe cases, almost 30 to 40 bunch will be dried up. A portion of the bunches will be dried up, and this is more severe in Thompson seedless. We can see lot of shriveling, berry shriveling at the tip of this, and even at the time of harvest also they will dry, but they won't detach from the berries. So it will affect the market quality of the bunches. It will retain, they will still attach to the bunches even after harvest, but they will dry and wilt. So this is this typical symptom of the cluster tip wilting. So what are the reasons? Reasons for cluster tip wilting is excess crop. That's what, high crop load in relation to its maximum capacity, because for each crop there is some limit. Beyond that limit, vines cannot nourish such a large, huge crop. So excess crop is the reason, first reason for cluster tip wilting. And second one is insufficient leaf area, because each bunches needs sufficient leaf area. I think in Thompson seedless, it has been standardized that for 450 gram bunch, we require around 2650 centimeter square leaf area. If there is a lesser leaf area, the nourishment of the bunches will not be sufficient. The tip portion won't get sufficient nutrition, that is malnutrition will happen. They start drying, and more compact benches also leads to wilting. So that's why even after application of GA3 also, manual thinning is very important. So immediately after 2 dips, we should go for manual thinning by doing thinning also, we can reduce this cluster tip, and poor micronutrient management and calcium deficiency are the reasons for cluster tip wilting. So control measures is adequate leaf area. That's what I was telling. You should maintain proper source to sink ratio. So if the one bunch is there in that particular leaf, at least 3000 square meter area should be there, a square centimeter area should be there to nourish that bunch, and judicious management of micronutrients, especially calcium nitrate. And this thinning I was telling, no, immediately after, at the time of thinning, you should tip off this at least 1/3 of the bunch, because even though it is retained, they won't develop into full size berries. So you can see here, these bunches at the time of manual thinning, you should trim off this tip so that we can reduce the formation of cluster tip wilting. And adequate potash nutrition and maintenance of adequate soil moisture during growth, they can reduce the cluster tip. And the last one important disorder is bunch stem necrosis. This also you are seeing here and there. This is one of the, because in a very good developed bunches, here and there, some of the branches in the bunches, they will start necrotic. I think many people, if you are involved in grape cultivation, some farmers are there, they must have witnessed this. There will be wilting of some of the necrotic spots will be seen in some of the bunches, but other this one branches will be normal. You can see only in portion of the stem of the bunches we can see the necrosis. So in this affects fruit set and fruit ripening. It can be either early or late. So this we can classify as when at the fruit set stage or at the bloom period, if the necrotic appears, it is called as early bunch necrosis. And if it occurs at the late period, it is late bunch stem necrosis. Both are very economically important, because this necrosis, they will reduce the yield drastically. And this is this is also considered as one of the complex physiological disorder, because the exact reason is also not known. Sometimes we will confuse it with a late downy disease, even though if there is no downy also, sometimes we can see this kind of the disorders. It affects vineyards worldwide. This is the problem in most of the table grape varieties. So, but no pathogens, that's what I was telling. No pathogen was found to be involved to cause this condition. Certain weather conditions and vine nutritions, the nutrient content in the vine may be the some of the predisposing factors for bunch stem necrosis. So, what are those predisposing factors? Are low temperature and high humidity during flowering stage, that is bloom stage, and excessive rainfall are some of the at the veraison are some of the problems. During the early bunch stem necrosis, low temperature and high humidity are the causal reason, are this one environmental conditions. And after veraison, that is for late stem bunch necrosis, excessive rainfall during veraison may be the cause. And imbalance between calcium and potassium and low levels of nitrogen are the other causal reasons. And unaffected portion of the cluster develop normal fruit. That's what I was telling. Within the bunch, one portion will be necrotic, another portion will be normal. So developing management practice is very complex, and it varies from vineyard to vineyard. Some of the potential causes are previous studies, many worldwide, they have attributed this to magnesium, calcium, and nitrogen imbalance in the soil, and cooler than average rainy weather during ripening. So these are the predisposing factors. So control measures are too much nitrogen causes excessive vigor, which causes again bunch necrosis, and overuse of nitrogen fertilizers, and judicious nitrogen management practice should be followed. And some of the researchers have also inferred that sugars in the storage organs in table grapes, they lead to low levels of sugars due to more nitrogen accumulation during cold weather conditions. So in this period, magnesium sulfate application also helps in overcoming the bunch stem necrosis. If you are giving magnesium, better to give magnesium sulfate in the soil application, or also in fertigated some of the soluble grade magnesium sulfate can be sprayed to the foliage, or we can give the magnesium sulfate to the soil. So through this we can control to some extent bunch stem necrosis. And one more important disorder is berry cracking. That this at the time of harvesting this will cause, and some of the varieties like Sharat seedless, Fantasy seedless are very susceptible for this. Mainly this is because of the property of the skin. Skin elasticity will be less in such varieties, and whenever there is a sudden rain at the time of harvest, there will be engulf of water into the berries, and the berries will start cracking. So they will involve interaction between environmental conditions and integrity of berry cell wall. So some fruit cracking, this because this is mainly because of the temperature and irrigation related problem. You can see here like this on the berries, you can see a lot of the cracks. Immediately after cracking, because of this moisture, some of the secondary some of the fungus like secondary pathogens will attack, and they will cause rotting of the berries, and ultimately marketing value will be reduced. So this is very important disorder because it will cause huge economic loss, because it will come at the fag end of the crop, that is at the time of harvest. So how we can control this is important strategy to avoid cracking is careful water management. So proper water management should be done, and use of some anti-transparent and micronutrients. So and calcium spray, they can increase the cell wall thickness in the skin, and they can improve their elasticity. And berry cracking can also be restricted by spraying the calcium compounds and growth regulators of GA3. And in a normal where in temperate climate like North Indian conditions, you should hasten the pruning, means we should prepone the pruning, so that we should avoid harvesting at the rainy season, so that berry cracking can, because this is the major problem in North Indian conditions, because their harvesting coincides with monsoon. So early ripening varieties like flame seedless, perlite, beauty seedless, they should be pruned early, so that they can be harvested before monsoon. And so another important is rudimentary panicle. These are all the small other disorders. You can see immediately after forward pruning, normal cluster won't develop. There will be small clusters. In each bunch, only 5 or 6 berries will be there. This is because of the imbalance in some of the this one hormones like auxin, cytokinins, and GAs. If there is a more GA, there will be turning of the normal berry bunches into foliage. That bunches won't develop into full. There will be some tendril, along with the tendril some rudimentary panicles will be there. So too many fruiting canes, too many growing shoots, water stagnation, prevention of chilly weather, these are the some of the causes for rudimentary panicles. Control measures like limiting the number of canes, that's what I was telling. Shoot thinning, 5 per meter square in Thompson seedless and 10 per meter square in Anabichaei, allowing only 2 shoots to grow on each cane, which can reduce this formation of the berry and maintenance of the adequate leaf area. So these per cane is very important management practices. Avoid water stagnation during flowering stage. So these are the some of the control measures for rudimentary panicles. So these are so today I covered mostly some of the important physiological disorders which will drastically reduce your post-harvest quality also, and also economic yield of the crop. So good management practices to overcome these physiological disorders should start from right from the training, pruning, and also adjusting time of the pruning. Good management practices, canopy management practices like shoot thinning, shoot positioning, bunch thinning, berry thinning, and good balance between source and sink. That is the one of the major thing we have to follow in grapes. Good canopy management involves proper source to sink ratio. That's what I told. In many of the disorders like water berries, pink, short berries, even your fruit cracking, immature canes. What I discussed all this past 1 hour, these are mainly because of the variation imbalance in the source and sink ratio. So first and foremost thing is you develop a proper source strength. That means you should maintain adequate leaf area and try to minimize the yield according to the source available. Or else what happens? Excess sink area can exhaust the reserves in the vines and also in the canes. That will reflect in the next year. That's why you should maintain the proper source to sink ratio with the good management practices, and then adjust the pruning time wherever possible. In the extreme conditions, adjust the pruning time so that we can overcome the some of the climatic factors, adverse climatic factors, where if you can anticipate well in advance, so that you can adjust the timings of your pruning, so that to some extent we can reduce those physiological disorders. So these are some of the important physiological disorders that I can briefly tell you in past 1 hour. I think I hope everybody, at least you come to know about some of the physiological disorders. What are all the important management practices to be followed to overcome such physiological disorders? And that's all for from my side for today's class. If you have any questions, you can please ask. Thank you.
Thank you, sir. Thank you. So any participant having the question, please ask to sir, or they can put the in chat box also the question.
Hello.
Yeah, tell me.
Good afternoon, sir.
Good afternoon,
sir.
good afternoon, tell me.
Thank you so much, sir, for this wonderful presentation.
Yeah.
Sir,
Thank you.
I'm from Kashmir. I'm Dr. Arusa.
Yeah, please.
I'm from Kashmir.
Yeah,
So generally, we predominantly have a variety called Sahibi.
Sahibi,
It's a seeded. So it's a seeded variety over here.
Yeah. Chima Sahibi.
No, it is just a simple Sahibi only.
Okay,
not
okay.
Kali or
Okay.
a single Sahibi.
Okay.
So it has been introduced long back by the late Maharaja.
Yeah, I
Kashmir.
know.
So now it is the most predominant variety over here.
Okay.
But the farmers, sir, they are having a very severe concern about the hen and chicken disorder in their vineyards. We are trying to manage it, but we are not able to manage it to that extent. Because it's a seeded variety, GA is not
required.
The variety is not responded to GA and all.
Okay.
Boron we are applying, pruning we are doing. But still, sir, can you please suggest that what are the measures that we can take to curb this disorder?
What is the cultivation practice there? Whether they are training properly? Which method of training they are following in cultivation? Whether
Sir,
they are
generally the training system is predominantly the bower.
Bower, okay.
It's a cane bearing variety. So need to be pruned at 8 buds.
8 buds, yeah.
Yes.
Okay.
So they are maintaining that.
What is the yield level? Means this one per vine, how much canes bunches you are retaining?
Sir, the vines are around 30-35 years old.
30 to 35 canes, shoots, bunches you are retaining.
No. Sir, the vines are
age.
30. Age is 30 to 35.
And on each vine, how many canes are there?
Sir, probably it will be 200-300.
Now, because what happens, no, here, I think 30 years means just, can you do some corrective measures like that? Because, after 30 years, there will be formation of some dead roots here and there. So translocation of these nutrients from root to the, because I think I hope this 30 years means there will be lot of spreading of the bunches, and here and there some barrenness is there.
True, sir, true.
So if they want to forego just at the time of pruning, and you prune only once, no, there, that is single pruning, no?
Right.
Yeah, and during pruning, try to remove those barren shoots, because here, as I told, the short berries or ICAR-NRCGrapes disorder is also due to poor translocation of carbohydrates from source to sink, right?
Okay. Okay.
And how about leaf?
But sir, even young wines are susceptible.
Then you can try
Even the
at
young ones.
at the time of this flowering, you can try some oxygen compound spraying, ma'am. IAA,
Okay.
indole acetic acid. Huh? You can give
Yes, sir.
you
Yes,
can
sir.
give some 5 to 10 ppm IAA. You can spray at the time of pollination, and you are giving boron.
Okay. Okay. Sir.
Boron.
Yes, boron we are giving. Yes, sir, boron we are giving.
Okay.
So,
Okay.
what is your opinion regarding the boron application after harvest? Means in the autumn application. Is there some impact of post-harvest application of boron on this disorder?
Nothing will be there. It should be at the time of just cluster emergence, at the time of pollination, because boron is involved in where for pollen growth development.
True, sir.
So during that time,
So, but sir, there is also some literature. There's also some literature wherein they have used boron after the harvest. Means
after
autumn
the
application
harvest,
of boron.
whether it is soil application or spray.
No, spray only, sir.
Okay.
They are
But
spraying it on the plant.
after harvest, when you spray, and then you will prune, no?
Yes, true, sir. Yes, after that pruning is done.
After pruning, if it is done, it is okay.
So maybe then the plant is storing the boron for the next year crop.
No,
maybe
the quantity will be very less, no? Boron application will be, because the boron requirement is very either in 1 to 10 ppm is the optimum.
Yes. So here we are using at a concentration of 1 gram per liter.
Okay. Then you can try some oxygen compounds, ma'am. And some of the varieties by its inherent capacity, some are susceptible for this hen and chicken disorder. It is their inherent inbuilt capacity.
Okay. So means it's also genetic then?
Genetic also may be there, because some of the varieties, what I showed, no? Some wine varieties, whatever you apply, they have short berries.
Okay.
Some are genetically controlled. So you can spray some oxygen compounds next season, and you can see, because I have not worked in that variety. So based on the available literature, you can try some oxygen compounds.
Fine, sir.
Okay.
Dr.
Thank you so much, sir. Yes, ma'am.
Sir, I just wanted to know, like in winter pruning, how many buds you prune, at how many buds you keep for pruning? Or because
8 buds. 8 buds per cane.
That is cane pruning they are doing.
So
and two we are keeping as a renewal spur.
Because ma'am,
we have one time pruning, na, sir.
Yeah,
so
that's why you have to maintain both the canes, spur also, cane
vegetative
also.
and the fruiting ones.
Yeah. For next year, that will become your cane, right?
True, sir. True.
So you will maintain 50-50 or 60-40, right?
Right.
So it will be the spur will also be there and cane will also be there. So
right.
spray boron and other things, no? So maybe this spur, whichever is there, it will become cane during next year. They may get benefited with your boron application for next crop.
Right.
Okay, sir. Yeah. Boron application I am doing a lot.
Okay.
But still it's not
what
managing.
About zinc?
Yes, zinc I have also tried, but it showed the phytotoxicity, sir.
How much concentrate? 0.1% zinc you can give. 0.1, that is sufficient if you give in foliar spray.
Okay, sir. But I need to add then lime, na, sir?
You try these micronutrient mixtures along with slurry. You can give in drip irrigation.
Okay.
Here and once in a while you can give foliar spray, but soil application along with slurry you try. You conduct your experiments, some 3-4 treatments you only can give. I told about oxin, one zinc concentration, one boron, and zinc, both soil application and this thing. And immediately after fruit set, no?
Okay.
You try to do some thinning also. because
Okay, fine,
that may
fine,
not be
sir.
economical for your condition, but to
True,
overcome
sir.
this disorder, you can do some experiments yourself. You can try some
Okay.
manual thinning.
Okay, sir. Okay.
But some of the this one, no, even seedless varieties, seeded varieties, little concentration of GA will respond. Okay.
Okay.
So
Okay.
first you try to increase the rachis length by giving light GA, 5 ppm, less than 5 ppm you do. In one
Sir,
row you
I have tried GA also, but
okay.
it's also not responding. There was no rachis elongation, though.
How much GA you
Other
gave?
sir, GA.
What is the concentration of GA you gave?
Sir, I actually used 10 to 40 ppm.
No. You go for
at different
for
stages.
seeded varieties, you should go less than 10 ppm.
Okay.
Because
Sir.
already GA will be synthesized in, because I will give
It
red
is
globe.
there.
For red globe is also seeded variety. I give
True, sir.
5 ppm.
Okay.
It will give very good rachis elongation.
Fine, sir.
So
Fine.
you try below 10 ppm. Don't go for 40 ppm.
Okay, sir.
You try this season.
Okay, I will try. I will try,
And you
sir.
can take my number also, even NRC people number also, and you can give your feedback.
Yes, definitely, sir. I need your guidance, sir.
Okay, sure.
Thank you so much, sir. Thank you, sir.
Any other questions, please? I think, sir, questions are over.
Okay, thank you.
Because you have presented in very nicely, sir. So I think all are get good information. That's why they don't have questions.
Okay, thank you.
So, sir, thank you very much, sir. You have given your valuable time for this training program as a research person, and you have presented very well, sir. So, thank you very much for your.
Thank you. I thank you, all of you, organizer and also director and the organizing secretary, for giving this opportunity to share my experience on physiological disorders of this management. Thank you.
Thank you, sir.
Satisha sir. Good
Sharmishra,
afternoon, sir.
How are you?
I am fine, sir. I joined a bit late, so half of the lecture I could attend, but I just wanted to wish you good afternoon.
Even I started because there was some problem in joining.
Yes, sir.
So I started late. So
Yes,
I ask your pardon for every participant because 15-20 days I was also late.
Yes, sir.
Okay.
Thank you, sir. Thank you.
Thank you. Okay.
Okay.
Dr. Yadav, sir, have you joined?
Yes. Can you hear me?
Yadav, sir?
Can you hear me?
Yes, sir, you are audible. So, participants, I will request you to be with us for the next lecture. That is on your integrated pest management in the vineyards. So this lecture will be delivered by Dr. D.S. Yadav Sahab. He is a senior scientist in NRC-Grape, agriculture entomologist. So, sir, I will request you kindly deliver your lecture. Please, sir.
Wait. So, thank you, Dr. Nikumbe. Who are the participants?
Sir, here most of the participants are MSc PhD students. Then few are the assistant professor from SAUs and few participants from the agriculture officers from the state agriculture departments. So it's one kind of the blend. So we need to think about the students also. Basic concepts or whatever is there. Okay, sir.
And anyone, most of the participants, they may not be having any experience in grapes.
Yes, that is.
So basics we will cover. Okay, in very brief, we'll discuss about the pest management in grapes. Good afternoon, everyone. Shall I share my screen?
Yes, sir, please.
Is my screen visible?
Yes, sir, visible.
Okay. We have about 6, 7 very important pests which are affecting grapes. Some minor pests are also there, which we may not be covering in this lecture. So we'll start with the first one. We have two prunings, April pruning and October pruning in grapes in the peninsular India. So after each pruning, there are buds which sprout. So we get first pest which attack on that is the flea beetle. This pest attack on the emerging sprouting buds and young shoots, and later on they can also cause damage to the young berries, peduncle, and the young stems also. This pest is nowadays becoming very serious, and farmers are reporting that this is not getting controlled easily. The thing is, the insecticides which are registered for this, which are the imidacloprid and lambda-cyhalothrin, they are not giving a quick result at the time of bud sprouting. And another thing is that why farmers are not getting control of this pest very effectively is that this pest is nocturnal in nature. It avoids sunlight, and at the time of evening and early morning also, it starts causing the damage, start feeding on the sprouting shoots. Because there is no canopy, and during the daytime, they keep on hiding under the debris or anywhere else where they can find the shadow. As they are feeding nocturnally, if they are spraying the insecticides during daytime, they may not be very effective. So what we suggest is spray at the time when the flea beetle are more effective, more active, mostly during 7 o'clock in the evening to 9 o'clock in the night. What are the insecticides which are effective? Imidacloprid, lambda-cyhalothrin, they are not giving the quick result. But there are other insecticides which are registered for thrips like spinosad, spinoteram, fipronil. There is one insecticide which is under registration, which is a combination product, imidacloprid plus beta-cyhalothrin. That is also giving very good and very quick result. That is the probably it will get registration within a year or so for the grapes. So these are the insecticides which we can use. And for detailed list of insecticides, you can refer our website, which keeps on updating regularly. And under for farmers section, you'll find two annexures, Annexure 5 and Annexure 9, where you can get the recommended list of insecticides. Also, for with this training manual, you'll also get the recommended list of insecticides, as well as the write-up on how to manage the major pests in grapes. Next is thrips, which is a major pest. Thrips, the species what we get here is, I forgot to tell you what species of flea beetle we have in grapes. This is Scalodonta strigicolis. This is a monophagous pest, and mainly it causes damage only in grapes. But sometimes we have found that it was feeding on pomegranate as well. But the damage was not significant, and not much population was found on the pomegranate. Otherwise, it is a reported as a monophagous pest. Thrips, the major species we have thrips is Cytothrips in grapes, and any succulent plant part which is above the ground, it can get affected by the Cytothrips. It can cause damage to the young shoots, young leaves, young stems, peduncle, young flowers, young berries. Rachis also. Any plant part which is succulent and above the ground, it can cause damage. First photograph, what you see here is pre-flowering inflorescence. If the thrips population is too high, it can even burn the whole inflorescence, and the yield is affected. In the middle photograph, you can see here that this is a pre-flowering bunch, in that before flowering, the damage was done on the outside of the flower. So probably this is calyx where the damage has been done. And when we controlled the thrips and this flower opened and the new berries were set, that time there was no symptom on the berries. All the berries were healthy. So, but once there is a damage on the young berries, we cannot do anything. And when the berries young berries after the damage, it is the scarring damage which thrips cause on the berries. And the berries, when they grow up, they develop, we can see the scarred symptoms. These photographs, first row, last photograph you see, these are the symptoms which we see on the grapes. And in market, whenever you see the grapes, these kind of symptoms, you can find out that these are the thrips berry scarring symptoms. Leaf cup, this is the first row, second photograph, is the symptom when the infestation is not controlled. These cupping symptoms you see in the leaves. Even the young stem, third row, second photograph you see, the scarring also on the stem is there. So thrips is a major pest, which is a very regular pest, and getting difficult to control as we require more and more insecticide applications. The best insecticides which work on thrips in grapes are spinosad, spinetoram, cyantraniliprole, fipronil, imamectin, benzoate. These are the insecticides which are very effective. Lambda-cyhalothrin, though it has label registration against thrips, but it is not able to provide any control of the cytothrips as of now. So we don't recommend lambda-cyhalothrin for the thrips. This is about thrips. Mainly what we do is insecticides we use for the management of thrips. One thing more what we can do is this, that after the berry setting, if we remove the excess shoot, then it can reduce the thrips population. So insecticides and removal of the excess canopy are the management options which we can practice. How do we know that how much thrips is there in the vineyard? So the monitoring technique is like this, that we take one piece of paper, white paper, and tap the shoot over the paper. That's how we can count the number of thrips fallen on the paper. And if they are more than 3 to 5, depending on the crop stage, we can consider that they are higher in number and we need to go for the insecticide interventions. One more thing, thrips population on the canopy varies during the daytime. Early morning hours and the late evening hours and even the night, you see the less thrips population. But during the daytime, you see the higher thrips population on the canopy. So time of monitoring is also important, that how much thrips is present. So best time is daytime when you monitor for the thrips population in grapes. One more pest which is important is leafhopper. What species we get is Ameresca bigotula. The major damaging period we have for leafhopper is from the 15th of September to 15 December. Majority of leafhopper population we get in the month of October and November. And how do we control it? The best thing about controlling leafhopper is this, that it is attracted towards the light. During the evening hours, if we install the light traps outside the vineyard, majority of the adult leafhopper population, they'll get attracted towards light trap and they'll die there. If insecticide application is necessary, what we can do is we can install a white color light source behind the tractor. What it will do while spraying at night, when we turn on the white light behind the tractor where the insecticide is being sprayed, all the leafhopper, they'll become active and come near the light. And the insecticide will directly go over the leafhoppers and it will kill them. So this is how we can improve the efficacy of insecticide which we are spraying for the leafhopper. The only insecticide which is registered for leafhopper is clothianidin by the soil drenching. But soil drenching was mainly, it was for the mealybug for which it was registered, but somehow it also controlled to some extent, not very effective. Leafhopper also, clothianidin as soil drenching, but that is not a recommended practice exclusively to control the leafhopper. The insecticides which are registered in grapes for use in grapes, but not for the leafhopper, they are better options which can control the leafhopper, such as lambda-cyhalothrin, very effectively, it works. Fipronil, imidacloprid, clothianidin, but as a leaf foliar application, it controls the leafhopper very effectively. Buprofezin, this also gives the very good leafhopper control to us. So the key here is that if you want to control the leafhopper, we need to spray during the night time, after the dusk, 7:30 p.m. in the evening. And we install the light behind the tractor and spray these effective insecticides, and leafhopper can be controlled. Next important insect is Spodoptera, which is the major period of activity of Spodoptera. Mainly we have Spodoptera litura, Spodoptera frugiperda sometimes we get, but sometimes mixed population also. But the major species we have is the Spodoptera litura. How do we control Spodoptera? Spodoptera, it can cause damage to the leaves. Young buds, when at the time of sprouting, like flea beetle, cause the damage. Even on the bunches, Spodoptera can cause the damage. How do we control Spodoptera? The best management of Spodoptera is installing the light traps outside the vineyard. You may not need to spray any insecticide if you have installed the light traps. It is effective for the Spodoptera. It is effective for the leafhoppers as well. So both these pests we can control using the light traps. We should not install light traps within the vineyards because it may attract the pests from outside into the vineyard. And the plants which are near the light trap, they may get a higher infestation of these pests. So we should install light traps outside the vineyard, not inside the vineyards. Another important species what we have is mealybug. Mealybug can cause damage to different plant parts. At the time of sprouting, it can cause damage to the young shoots where it can cause the malformation. It can feed on the young leaves. It can feed on the suckers and the buds, sprouting buds on the main trunk if they are there. Later on, when the sugar is moving towards the berries, that time it can go to the bunches and it can infect the bunches as well. At the time of harvesting, this is the major pest which is causing the damage in vineyards, and it is a very serious pest and very difficult to control pest. How do we control the mealybug? It is a year-long process about managing the mealybugs. It is not that whenever the mealybug is seen, only then we take the measures. After each pruning, what we suggest is wash the whole vineyard with the whole plants, all the plants with the buprofezin, which is a registered insecticide for mealybug. As buprofezin is non-systemic in nature, it controls the mealybug by contact action. So at the initial period, mealybugs are feeding under the loose bark on the main trunk and the cordons. After October pruning, what we suggest is you remove the loose bark from cordons and the main trunk. After that, wash the main trunk and cordons with the buprofezin to control the mealybugs. After this one wash, monitor the vineyards after every 10 days. And whenever you see the mealybug infestation, you do the spot application and kill the mealybug itself. Why is that? Because mealybug in grapes has clumped distribution, patchy distribution. So initially you will see only few plants which are infected with the mealybug, and later on it keeps on spreading in the whole vineyard. So every 10 days, if you are doing the monitoring and controlling the mealybug at initial stage itself, it will not spread to the whole vineyard, and you'll be able to achieve better mealybug management with the less cost. Another thing that this picture shows that the bunches which are touching main trunk, cordon, wire, or cane, these bunches are having higher mealybug infestation. So at the time of April pruning, after April pruning, we should tie the shoots in such a way that all the bunches are in one line and hanging down without touching anything else. So these bunches can be easily covered by the insecticides It will not provide any hiding space for the mealybugs, and mealybug control can be achieved in better way. Another major pest which is difficult to control is stem borer. Stem borer species what we have are three different species of stem borer in grapes. First is Stromatium barbatum. Stromatium barbatum is a pest of old vineyards. It can cause only damage only to the dead wood, not to the live wood. On dead wood, when it is feeding like termite. It turns the whole stem, main trunk and cordons into the fine powder. While feeding on the dead wood, it also causes damage to the live wood, and the productivity of the vineyard is reduced within 2 to 3 years. As this is a pest of old vineyards, 7 or more year of vineyards are infected by the Stromatium barbatum. And there is no other method through which we can, there is no method available through which we can control the larva, because larva is inside the gallery. Gallery are tightly packed. There is no exit or entry hole for the larva. And you cannot apply any systemic insecticides also. And this larva is so resistant to insecticides that 20 times dose of any recommended dose also, it is not getting killed. Life cycle of Stromatium barbatum is at the time of beginning of rainy season. Adult cuts makes the exit hole, comes out. This black color ceramicid beetle is the adult of Stromatium. It comes out, it lays eggs under the loose bark, over the bark, anywhere in the vineyard, and larva direct bores inside. So last photograph here in third row, you see that there is a very tiny hole through which the larva entered inside. After that, the larva remains inside and keeps on feeding inside. The only stage which we are able to kill is adult stage. But we cannot spray so much insecticides throughout the rainy season. That's why practically it is not possible to control the adults also. Adults are attracted towards the light traps, but light traps are not effective enough to control or provide the management of the Stromatium barbatum. What we have found out is that neem has repellent effect on the Stromatium barbatum. So after the rains, if we create the smell of neem by doing the soil drenching of the neem leaf extract, or spraying the leaf neem extract, or hanging the neem leaves around the vineyard, so we can to some extent reduce the entry of Stromatium barbatum adults inside the vineyards. So it will reduce the egg laying, and the infestation level of Stromatium can be reduced in that case. Another thing what we can do is the majority of the egg laying happens under the loose bark. So at the beginning of the rainy season, if we remove the loose bark from the plant, it will provide less shelter to the adults as well as the site for the egg laying. So that's how we can also reduce the egg laying by the Stromatium barbatum. Keep in mind, all these practices are to be done in vineyards which are 7 years or older than the 7 years vineyard. Why this? Because when the vineyard starts becoming older, it starts making the dead wood. And this is the pest of dead wood, and it doesn't get attracted towards the live wood, and it doesn't infest live wood also. But while feeding on the dead wood, it can cause damage on the live wood and reduce the productivity of the vineyards. So in essence, we can kill only the adults, and we cannot control or kill effectively adults also. But we can use the repellent effect of neem to reduce the infestation of Stromatium barbatum. If the productivity is already 30 to 40% less than the potential productivity, in that case, what we suggest to grape growers is this, that your vineyard is already old and economic lifespan has already passed. What you do is you regenerate the existing vineyard by grafting on the existing rootstock there in the vineyard, or you replant the whole vineyard afresh. So that's how you manage this pest. Next important stem borer species we have is Dervisha kadambi. This is a Lepidopteran stem borer which we have in vineyards. Initial symptoms of infestations are in the month of June, July, August, and beginning of September, when the larva is feeding under the loose bark. No external symptoms are seen except that loose frost is and webbing is seen protruding outside the bark. Upon careful examination, you see the larva is feeding under the loose bark. In the month of September and onwards, it starts entering inside and going inside the trunk. Once it is inside, it makes galleries in the trunk, and productivity is reduced from the first year onwards. Once it enters inside and make the galleries, there is no way we can control this stem borer. The only way we can control stem borer is during the months of June, July, August, and somewhat in September, when the larva is feeding under the loose bark. We can control this insect. This pest is also a pest of teak plantations. For last many decades, it is a severe causing huge damage to the teak plantations. They have not found any management of this pest so far, any effective management. They are working on pheromones, mostly in Kerala, to manage this pest. This pest has also been found in the pomegranate, where there is no solution. The only advantage in grape we have for this pest is that at initial stage it is feeding under the loose bark, which is not the case in case of pomegranate and the teak plantation. So it is easier to control in case of grapevines. This larva has a habit that when you touch this larva or remove the bark, it will fall down. And just by removing the loose bark, we can control the majority of the larva because it reduces the shelter for this larva, and the larva gets eaten by the predators in the vineyards, present in the vineyards. So just by removing the loose bark in the month of June, July, and August, whenever this larva is feeding, we can control this stem borer. And another thing, that if you want to spray any thing for this, and you think that just by removing loose bark, it is not giving effective control, what we can do is we can go for the metarhizium entomogenous fungus application on the main stem and the cordons where it is feeding. And within 3 to 4 days, metarhizium provides mortality of this larva. It is a rainy season, humidity is already high. Metarhizium works excellently well. So removal of loose bark and application of metarhizium works wonders for managing this pest. Last pest we have is stem borer, which is conventional stem borer, Psilostrona scabrator. The first symptom what we see of this pest is intervennal chlorosis. It starts from the month of December. When we go near that plant which is having intervennal chlorosis, we see that some frost has come out. And when we go near the frost and remove the frost, we see there is a hole on the plant. When we tear apart the plant from the months of December to April months, we can see the larva feeding inside, and there is a gallery. Mostly sometimes 2 or 3 larvae are seen in one plant. Adults, they come out in the rainy season. They feed on the bark of the canes, the tender bark which is there. They lay egg inside the stem. They cut open the stem, they lay egg inside, and cover it with some gelatinous substance which gets hardened over time. So we cannot control its eggs. Adult emergence period is very long. From June month, it can keep on emerging until December. We cannot target adults for the management. Adult, they get attracted towards the light trap, but light traps are not effective in managing the Xylostrona adults. How do we control the Xylostrona is by monitoring. We know that from December month, it starts feeding and removing the frass. During that period, what we can do is at the initial appearance of the frass, we can every 10 or 7 days, we can monitor the plant. And whenever there is a initiation of frass removal, the larva is feeding near the hole only. It has not made the galleries deeper inside the stem, and it is there only. With the help of scissors or screwdriver, we can tear apart the hole and we can remove the larva and kill it. So that's how we can control the Xylostrona scabrator. There is no need of any insecticide application, no need of any other method, and it is very easy to control by just tearing apart the larva whole and removing the larva outside. So these are the major pests what we have in grapes, and in brief we have covered. You have any queries or any questions regarding the pest management, you may ask.
Good afternoon, Dr. Yadav.
Yeah, good afternoon, ma'am.
How are you?
Yeah, good. Who is this?
I'm Dr. Arusa from SCOST.
Yes, ma'am.
So I just want to ask that is it necessary that whenever we have the pests in the vineyard, we are supposed to spray? Or we need to take some prophylactic measures at regular intervals, maybe 15 days intervals, so as to curb the, and so whatever pest is going to come on the crop?
Not necessary that, but there are certain insects for which we have only chemical management. Like thrips, we don't have any other management option available to us. Flea beetle, we don't have any other management option. Like we mentioned that for leaf hopper and for Spodoptera, only installing the light traps is effective, and we don't require any insecticide applications if we have installed the light traps outside the vineyard. As light traps, we mentioned that they are not effective for the stem borers. We can use for monitoring, but not for the management. They are not effective there. So for like manual management, removing of loose bark for the Dravisha Kadambi and manual removal of larva at the initiation for Xylostrona are very effective. So these are the alternate methods we have. And prophylactic, like we mentioned that light traps we can use for caterpillars and leaf hoppers. And for mealybug, what we have suggested that after each pruning, we wash the whole plant with the buprophagin. So that is a prophylactic measure. And every 10 days, we can keep on monitoring, and with spot application, we can manage the mealybugs. So these are the, that's how we do that. For different pests, we have different strategies.
So does blister mite also come there?
No, ma'am. We have Tetranicus mites here, not the blister mites.
But the symptom you showed of thrips, not the leaf symptom.
The
leaf, one of the leaves were there. It was showing the blister-like appearances.
That was mainly
Can you
leaf
check
cupping.
that?
Yes, ma'am. That was leaf cupping due to the thrips infestation.
Okay, okay. Thank you so much.
Yes, ma'am. What are the pests you have in Srinagar, ma'am?
Mostly the flea beetle and the blister mite.
Blister mite, okay. Any other queries? Any other questions? I kept the lecture short only because my throat is paining. I have throat infection. So if there are no other questions, I'll stop now, and I request organizers to take up from here.
Yadav sir, thank you for the presentation. I think I'll just talk to Prashant sir about this.
There is one question in the chat. Is there any biopesticide for controlling mealybug and ants? From Dr. Radha Jadhav. Metarhizium works very well for mealybug as well as the ant. We can use Beauveria bassiana. Beauveria also works for mealybug. But metarhizium is very effective against mealybug, and it can kill both ants and mealybugs. There is one question. Please tell us about Stenius griseter, grapevine girdler. Mr. Kalbor has asked one question. When I was in BSc, MSc, and PhD, the only pest which we were reading in grapevine was girdler beetle. When I came here in Maharashtra, joined as a scientist, I found out that the girdler beetle is not a major pest. It is one of the minor pests with rare occurrences. Girdler beetle can infest vineyards which are new vineyards. Only up to 2 years of the vineyards they can infest. Mainly 1 year old vineyard, after the grafting, it is causing the major damage. In the months of October, September, October, mainly October, sometimes September and November also. It is very easy to control stem girdler. How? That new vineyards farmer is observing that there has been girdling which was done. Next night at 9:30 in the night, if you go to the vineyard on the same plant which the girdling was done previous night, you can see the girdler beetle. If not the same plant, nearby plants, you can see the girdler beetle sitting at 9:30. You can catch girdler beetle easily, it won't fly away. And you can kill by manually by any way if you like. So that's how you kill the girdler beetle. What damage it causes? We have done extensive surveys for the girdler beetle, and we have found out that upon girdling, it is a wound damage which is on the plant. If the plant is not cut in 2 parts, then what is there? That if you apply some disinfectant like Bodex mixture or then cover it with the cow dung, the plant will heal on itself, and within 15 to 20 days, it will heal completely. If the plant has been cut into 2 parts, the upper part will die. What we can do is we can cut the, prune the live part 2 inches below the cut, apply the Bodex mixture above it, and apply the hydrogen cyanamide. New shoot will come, and it will we can again raise that, and we can make the new plant. That's how we can heal the plant, and there is no need to apply any insecticide for the girdler beetle. Only manually we can kill, so that the new plants are not damaged next night. More questions. Any grape nematode related management? In grapes, nematode is a problem only in vineyards where grapevine are raised as on root plants. In grapes in India, we are mainly grafting the grapes on the rootstocks. Dogris 110R are the major rootstocks which are resistant to nematodes, and we don't have therefore any nematode problem in the grapevines. So that's it. That's all. Okay, Sarmishta madam, over to you.
Yes, sir. You done, sir?
Yes, ma'am.
Okay, thank you, sir. Thank you so much. Thank you, sir, for your valuable time and lecture. So, Holkar, sir?
Yes, I'm
Sir,
there.
probably we'll start with your lecture, Adaptive Spraying Technologies and Effective Chemical Solution for Disease Management in Grapes.
Okay.
So, sir, you can please start your lecture.
Okay. Slides are visible?
Yes, sir, they are visible. Slide show पे कर दीजिए, sir.
Yes. Now is it okay?
Yes, sir. Yes, sir, you can start, sir.
Am I audible?
हाँ वॉल्यूम थोड़ी मेरे में कम आ रही है, but I don't know about the participants, but
Okay, okay. I will
वॉल्यूम थोड़ा
increase
बढ़ा
the volume.
Yes, sir.
Now is it okay?
Yes, sir. Yes, sir.
Good afternoon, everyone. So, the adaptive strategies will be in the second lecture. First is the biointensification for disease management in the grape. So, good afternoon, everyone. This is Somnath. Today, we'll talk about there are different diseases in the grapevine and how the diseases are managed by different biointensification strategies. So, as we know all that the grapevine cultivation in India is an important horticultural crop, which is covering an area around 161,000 hectare, which is cultivated in three climatic conditions, that is tropical, subtropical, and temperate. Most of the grape cultivation in India is targeted for the table grapes, followed by export, resin production, and winemaking. So in 2022-23, India had exported around 2,67,000 million metric tons of grapes to the world, which is costing around 313 million US dollars. So this is export potential crops. So there are the different states of grape production in India. Although there are more than 10 states which are cultivating the grapes for the commercial purposes. But the area and production, as far as area and production is concerned, the Maharashtra, Karnataka, and Tamil Nadu, these three states are contributing nearly 80% of the area and production. So in grape wine, there are the major biotic constraints. So now we have listened about the major pests of the grape wine, and now is by Dr. Yadav sir. Now we see the what are the different diseases in grape wine. So and how the diseases are affecting the quality production in grape. First and foremost important disease is the downy mildew, which is causing around 80% crop loss. The second most important disease is the powdery mildew, which is caused by the fungus and Sinula nictator, which is causing around 80% of the loss in the grape. Anthracnose is the third most important disease in the grapevine. In India, it is caused due to Coletrichum gliosporioids. The severe infection may lead to 100% crop loss. This year, the anthracnose and the bacterial leaf spot disease has damaged the crop severely in certain areas of Sangli and different places. So due to the untimely rains which is followed by the different climatic conditions, rust is one of the minor diseases now becoming the major one due to the environmental conditions, weather conditions which are favorable for the occurrence of the rust. Post-harvest diseases, which are causing around 10% crop loss. Initially, we will see what are the different major symptoms of the different diseases in the grapevine. This is the downy mildew disease, which is the during the fruit pruning season. Downy mildew is one of the important disease till the 50 days of the fruit pruning, because the downy mildew infection, there are the under severe condition, it causes the huge losses to the grape growers. So we need to be very much cautious for management of the downy mildew disease under fruit during fruit pruning season. So these are the different symptoms which are appearing on the lower surface of the leaves, on the upper surface of the leaves. Oily spots which are occurring on the upper surface of the leaves, whereas the cottony cushion-like growth which is appearing on the lower surface of the leaves. In different flowering stages, carrot green stage, these are the different symptoms which are affecting the grape production. There are the different stages of crop growth where downy mildew is severely affecting the quality production of the grape wine, that is the cap fall stage, berry setting stage. If the proper preventive as well as the curative sprays were not undertaken in that situations, there would be a severe crop loss in the grapes due to the downy mildew occurrence. During the flowering stage, there if there are the conditions which are rainy conditions, which are cloudy weather conditions, which are favoring the occurrence of the downy mildew disease, and these are the symptoms when berry development stage is there. Second most important disease is the powdery mildew disease. Powdery mildew disease is becoming major constraints in the grape production during the berry setting stage till the harvesting stage of the crop. So the lay or inoculum of the disease which is on the vines, which is leading to the infection in the new leaves, in the older leaves, that is the whitish to grayish powdery mass which is appearing on the upper surface of the leaves, which is followed by the appearance of the powdery mildew disease on the canes and subsequently affecting the quality of the bunches. So this is the grape vine cultivar, which is the red globe, which is severely affected due to the infection of the powdery mildew. So this is affecting the quality as well as the marketable value of the crop and leading to 100% loss to the grape growers. Third most important disease is the anthracnose, which is the disease of the rainy season. So young growing tips and leaves are affected majorly by the anthracnose disease. So at different crop growth stages, these are the symptoms. Short pole symptoms is a very peculiar and which is the identifiable symptoms of the anthracnose disease on the grapes. So lesions, which is followed by the lesions on the stems, bud necrosis, then berry rot, then flower necrosis. These are the different type of symptoms of anthracnose disease in the grapes. Very unusual symptoms of the anthracnose disease, which is not very common in the Indian conditions, but during the last few years, the symptoms of the anthracnose disease are appearing on the berries. And in this season, 23-24 season, we have witnessed Sangli area and many other areas. Those areas received the untimely rains during the berry setting stage. All the grape vineyards were affected by the anthracnose, followed by the bacterial leaf spot disease, which is caused by Xanthomonas campestris pathovirus. So this is these symptoms are governed by the. So these are the pictures we have collected the samples from the Sangli area. So and by the infection, because there are the many other factors which are responsible, which are the predisposing factors which are responsible for occurrence of the anthracnose as well as the bacterial leaf spot diseases. So we have collected the samples and identified the associated pathogens with the diseases. So Colitrichum gliosporides and Xanthomonas campestris viticola were identified as one of the causal agents of the berry rots of the this grape. So fourth important disease is the bacterial leaf spot disease. These are the symptoms on the lower leaf surface. We can see as the water sprinkled on the lower surface. These are the water-soaked lesions which are confined to the ventlets. On the upper surface of the leaf, these are the angular leaf spots which are restricted to the veins. So and after a certain period of time of occurrence of the disease, the leaves become necrosis and they get dried. So apart from the fungal, bacterial, and viral infection, bacterial diseases, there are more than 100 virus and virus-like species which were identified, which are infecting the grapevine worldwide, not in India, but this is a scenario of worldwide grapevine viruses. So they are belonging to different families and genera. So these are the different symptoms. So majorly all the viral species are associated with the few diseases. That is first and foremost important viral disease is the leaf rot disease, rugose leaf blotch disease, flake disease, and infectious degeneration and decline. So these diseases are complex etiology, having complex etiology because the infection of or occurrence of the diseases are caused by more than 2 or 3 virus species in a disease. That's why we are calling it as a complex etiology diseases. So these are the different viral pathogens which are associated for leaf rot diseases. Grapevine leaf rot associated viruses are there. Mottling and deformation, rugose wood. In case of rugose wood, grapevine viruses are reported. These variants are from A to M, and grapevine rupestis tempeting associated viruses. So you can see in the picture the rugose wood, that is the stem of the vine is becoming degenerated. And all the red blotch symptoms are also there on the lower surface of lower slides of the slides. So these are the, in nutshell, these are the different diseases, fungal, bacterial, and viral diseases. So for management of the diseases, we are focusing only on the integrated disease management approaches, because the biocontrol strategy alone will not suffice the purpose, because we need to talk about the integrated disease management strategies. So in this lecture, we will only focus, these are the different components of the integrated disease management strategy, starting from cultural practices, mechanical measures, use of chemicals, use of biocontrol agents, botanicals, resistant varieties. So use of biocontrol agent becomes very important, as we have heard from the director sir, that why biocontrol measures are important, because of the residue issues which are occurring in the grapevine, and because of the and second most important is the management of the different diseases and to reduce the residue level. So the biocontrol agents are from different niche, we can say. So these biocontrol agents are originated from different locations, the different plant parts, different soil conditions, that they are rhizospheric, isolated from rhizospheric soils. They are epiphytes, they are endophytes. So we'll see all the biocontrol agents. So before managing the disease, we need to understand what are the predisposing factors which are helpful, which are responsible, predisposing factors which are responsible for occurrence of the disease. So for occurrence of the downy mildew, there is a thumb rule, that is 10-10-24. So 10 mm minimum rainfall is required, or moisture is required, and 10 degrees Celsius temperature is required, which is lasting for 24 hours in the vineyard. So these conditions are causing the downy mildew occurrence in the disease, followed by the cloudy weather and all the different temperature conditions for different period of time. So which leading to the occurrence of the downy mildew disease. Downy mildew disease in the vineyard. So first and foremost important practice is the cultural practice. So the branches of the vine should not touch to the ground, because when there will be a rainfall, the rain splashes will be there, and the spores from the soil, they will be reaching to the vine and will cause the first primary infection. So we need to avoid the branches touching to the ground. By this way we can avoid the source of the primary infection and further spread in the vine. Most important criteria of the protection is the after fruit pruning or foundation pruning, we need to dispose the pruning material, the pruning all the leaves and the canes. We should destroy, because all the material will carry the inoculum of the diseases. So we need to conduct the clean crop cultivation, so that we can kill the overwintering pathogen or inoculum present on the pruned plant parts. So for that we need to store these, we need to dispose or need to collect these pruned material, and it should be sprayed with the biocontrol agents or any contact chemicals, and it should be used for the compost making, so that for the next season there should be, and it can be available as a compost for the vineyard. So early sprouts are occurring in vines, and these early sprouts are the harboring source for the occurrence for the downy mildew pathogens. So these early sprouts we need to remove by and for removing the early sprouts, because we need to keep the canopy even, and for that purpose we need to remove the early sprouts. And for removing the early sprouts, these activities should be carried out during the sunny weather, because it should not be cloudy or rainfall conditions in the vineyard, so as to avoid the spread of the pathogen. Fourth most important is the removal of unwanted leaves, because the canopy management plays a very important role. Canopy should be very clear in early stage of the crop when the fruit pruning is done. So removal of the unwanted shoots and basal leaves are important to avoid the development of the microclimatic conditions, so which are harboring the different diseases, downy mildew, powdery mildew, anthracnose. So if the canopy is very much dense, then it will be very difficult for the management of the diseases and to so as to cover the crop, and this will facilitate the microclimate development, and so management of the diseases becomes very difficult. So use of biocontrol agent is very important. So there are the different biocontrol agents are originated from different rhizospheric soils, epiphytes, endophytes. Epiphyte means those microbes which are appearing on the surface of the berries or leaves, and the endophytic microbes, which are we'll see in the later slides, the endophytic microbes. So these are the different biocontrol agents which are used routinely in the grapevine for the management of the different diseases and for reducing the residue. So trichoderma is one major biocontrol agents. There are different species of trichoderma: aspergilloides, afrohargianum. Then Bacillus subtilis is the bacteria, Ampelomyces piscicalis, Bacillomyces, Pseudomonas fluorescens. These are the different biocontrol agents. So you can pick in picture, you can see these are the different biocontrol agents: Trichoderma aspergilloides, Trichoderma afrohargianum, Bacillus subtilis, and Ampelomyces piscicalis. These biocontrol agents are available at our laboratory at ICAR-NRCGrapes Pune, and we are working on all the isolates. So these are used, Trichoderma aspergilloides is used for powdery mildew, downy mildew, anthracnose and other post-harvest decay. So afrohargianum is widely used for powdery mildew management and downy mildew or anthracnose. Bacillus subtilis is mainly used for the powdery mildew and residue management. Ampelomyces is mainly for the powdery mildew management. These are the different biocontrol agents. For any biocontrol use, there should be the spore count in a formulation. There should be 10 raised to the power 8 formulation spore count should be there. The temperature conditions varies for the different biocontrol agents. So under low temperature conditions and humidity conditions around 80%, these biocontrol agents acts very nicely under field conditions. So at higher temperature, these biocontrol agents may not and may not give the actual results so as expected. So for management of the powdery mildew, if we alternate the sprays with the sulfur by the Trichoderma aspergilloides, it controls the powdery mildew. So Trichoderma aspergilloides, if we go for weekly interval, the powdery mildew occurrence will be limited. Bacillus subtilis, the conditions will be 28 to 30 degrees Celsius, and at 80% relative humidity, it will act nicely. So the doses of the biocontrol agents will be 2 gram to 2.5 gram or 2.5 ml per liter of water will be used for the application in the vineyard. Ampelomyces piscicalis is one of the effective biocontrol agents against the powdery mildew, which is the and along with the Ampelomyces piscicalis, if we alternate our sprays with the chitosans, so most of the time when there will be a horizon stage and sugar accumulation stage, that time the berry cracking problem will be there. So if we alternate our sprays with the Ampelomyces and chitosan, so it gives the better control against the powdery mildew and berry cracking will be avoided. So for the better performance, we can alternate our sprays with the two agents. So ICAR-NRCGrapes Pune has developed two formulations. One is the Manjari Wine Guard, which is recommended at 2 ml per liter, Manjari Trichoshakti, which is a powder formulation, so which is sufficient for 1 acre. It is a 10 gram pouch, sufficient for 1 acre application. So starting from the foundation pruning or fruit pruning, so we need to wash the entire vineyard or entire wine cordons with the biocontrol agents for control of the powdery mildew, because the powdery mildew, downy mildew pathogen inoculum will survive on the different plant parts and in the soil. So we have to remove, we have to suppress the initial inoculum level, so that in the main season there should not be any occurrence of any severe occurrence of any disease. So we have to reduce the inoculum pressure. So for that purpose, we need to wash the wine or cordons or canes with the different biocontrol agents. Better go for trichoderma and bacillus with different doses. Trichoderma, if it is a formulation which is available in the market, we can use 5 ml or 5 gram per liter of water. So, and the volume of the water can be extended to 1.5 to 2 liter per wine, or and there are the formulations which are available with the NRC grapes, that is Manjari wine guard, which can be used at 2 ml per liter of water for washing the entire vineyards. So after these biocontrol agents can be applied frequently in the vineyard, so as to avoid the occurrence of the severe occurrence of the diseases. So starting from the 1 week before the fruit pruning till the harvesting of the 10 days before the harvesting of the crops. So biocontrol agents like trichoderma and bacillus will help in managing these different diseases. In vineyard, sometimes there will be conditions that entire vineyard cannot be pruned at the same times. So in that situation, if there is a time or gap of fruit pruning or foundation pruning, so we have to apply the nearby areas, nearby vineyards, those who are unpruned, with the biocontrol agents, so that we can avoid the further spread of the diseases from old vineyards to the newly or pruned vineyards. So we have to take care of the activity by spraying the biocontrol formulations. There are the different critical crop growth stages. At different crop growth stages, this is a very important crop growth stage. That is a bud sprouting stage. In this stage, if there are the conditions, that is cloudy conditions or rainfall is there, so there are the chemical strategies which are to be followed, so that the further spread of the downy mildew is to be avoided. So in this, if the conditions are normal, if temperature and relative humidities are normal, sunny weather is there, then the application of trichoderma is essential at this stage. Either it should be a drench or spray will be helpful. So at the 15 to 35 days of the fruit pruning, also the application of the biocontrol agent is very essential, either through the drip irrigation or through the foliar sprays. But the foliar sprays are essential in this stage, because these are the crucial crop growth stages which are affecting, which are sensitive for the occurrence of the downy mildew disease under field conditions in the fruit pruning season. During the 36 to 50 days after fruit pruning, so in that conditions, the flowering to berry setting stage conditions will be there. So in that conditions, if the weather conditions are normal, or no heavy rainfall or no cloudy conditions are there in that situations, so preventive application of biocontrol agent is essential. So application of the bacillus followed by the ampelomyces quiscalis should be applied by the foliar sprays to take care the powdery mildew or downy mildew occurrence and the anthracnose as well. And during the berry development stage application, at least 2 applications of either of these biocontrol agent, that is chitosan, ampelomyces, bacillus, or trichoderma, is essential to avoid the berry cracking occurrence of the, because the stage in this berry development stage, the occurrence of the powdery mildew starts when there will be a temperature rise in temperature. So we have to take care the application. We have to go for the application of these biocontrol agents for management of the powder mildew. 90 to 130 days, yeah, which is the from horizon to harvesting stage. At this stage, at least these sprays and drenching of the biocontrol agent, that is at least one spray of trichoderma, followed by the spray of bacillus subtilis, and alternate sprays of these two, bacillus and trichoderma, in the 90 to 130 days will 90 to 130 days will be helpful in different parameters, like reducing the pesticide residue, so reducing the inoculum pressure of the powder mildew, so and which may enhance the shelf life of the grapes. So we have to follow these biocontrol strategies. In brief, we'll discuss about endophytes. Endophytes, these are the microbes which are residing within the host, every host. In 5 minutes, we'll finish it up. So 5 to 10 minutes. So endophytes, these are the microbes which are residing within the host plants, which are either for the part of the host plant or entire host life of the host. So these are the symbiotically stay within the host plants. So these endophytic fungus or bacteria, they are having the different beneficial effects on the host plants. So these are the different application of the endophytes. They are imparting the plant growth promotion activity. They are helpful in the nitrogen fixation, increase the crop yields, pest disease resistance. They are potential biocontrol agents. They are helping in the phytoremediation and uptake of important nutrients in the vineyards. So these are the different different benefits of the endophytes application in the vineyards. So by keeping this view in mind, we have started the study on the application of the endophytes in the grape wine. We have isolated more than 200 endophytes and characterized around 120 fungal and bacterial endophytes, both based on morphologies and based on the molecular characterizations. The sequence information is also submitted to the NCBI gene bank. So, and we, apart from these, we have screened the formulations of the trichoderma, so which found very beneficial, which are highly antagonistic under in vitro as well as under field conditions. So we screen out the trichoderma asperillum endophytic fungi. So these are the different bacterial endophytes which are showing the antagonistic activity against the Polytrichum gliosporides, which is causing the anthracnose disease in the grape. So these both bacillus as well as the trichoderma formulations we have shortlisted, and we have developed the formulations under field conditions. We have applied these formulations for management of the powdery mildew. So this is a 22-23 year. We have conducted the trial on the grape wine cultivar red globe, and promising results obtained. So initially, before, this was the plot which was severely affected by the powder mildew, and so we have carried out these sprays. We have undertaken these sprays in the red globe variety. So, and in a span of 45 days, we have applied the 6 sprays of the formulations, bacterial as well as the trichoderma formulations, and obtained the promising results in management of the powder mildew. And in this time, we did not use any chemical fungicides for the management of the powder mildew. So, so as to, and in the case of the untreated control, there was a significant improvement in the management of the powder mildew disease under field conditions. Similarly, subsequently in 2023, we have carried out the sprays of the endophytic formulations in Sarita seedless, Sarita, and we have obtained the promising results for management of the powder mildew disease under field conditions. So this is these are the photographs at the maturity stage. So apart from these, we have recorded the unusual lip spot symptoms of the grape under field conditions. So we have carried out the identification of the fungal pathogen associated with the leaf spot disease. We characterize these pathogens morphologically, as well as molecular characterization was carried out, and we found out that the Fusarium equisetae species, which is responsible for causing the leaf spot disease of grapes. Similarly, the wilt disease is occurring in many pockets of the Maharashtra area, so which is leading to the sudden decline or death of the plants in the vineyards. So most of the areas from the Nashik as well as Sangli and Solapur, grape growers are facing the same problem of the disease. So we have collected the samples and observed the symptoms. So we found that when we split the root portion of the affected vines, so there was a severe reduction in the xylem. Brown discoloration occurred in the xylem portion. So, and from that we could cultivate the fungal pathogens which are associated with the disease, because these are the Fusarium species based on the morphological and colony characteristics. We have identified it is a Fusarium, but we have to identify based on the molecular characterization. So this in the first slide, you can see, firstly picture, you can see that V notch symptoms, which is a very peculiar symptoms of the which is reported in the different countries, which is called the wilt disease or decline disease. So, and the pathogen is growing across the nodes, internode nodal transmission of the pathogen is observed. So this is this disease is becoming a major problem nowadays. So we are suggesting the different management strategies to the grape growers. Initial stage, we have to whether if the symptoms are observed in the few vines in the vineyard, then we have to reduce the water application. We have to reduce the PGR applications, because these applications may lead to the increase in the disease in the other plants which are nearby. So we have to drench the these affected vines with different biocontrol formulations, and drench the along with the subsequent drenching is needed, because one or two drenching will not be sufficient, and through drip irrigation also, as well as through proper drenching should be followed by percolating maximum biocontrol agents to the root zone of the vines, so that the wilt disease can be managed in the early stage of the detection. So diagnosis of the disease is very much important, because most of the time what happens is that there should be a physiological disorder or the wilt problem. So whether we need solutions, because the root portions become dormant during condition in the problematic soils. So we have to identify the disease, diagnose the disease very properly, so that proper management strategies can be applied. So for management of the viral diseases, the integrated disease management strategies can be implemented. Although the management of the viral diseases is very difficult, so we have to have the which is having the disease or virus-free planting material with us, so that the management of the virus spread or decline in the vineyards is occurring due to viral problems. So we have to have these approaches. So in order to study the viral problems in the vineyards, we have to first detect the association, detect the viruses which are associated in different diseases. So in India at present, although there are more than 100 plant viruses which are affecting the grapevine globally, but in Indian conditions, roughly there are around 20 to 22 viruses which are reported based on the ELISA and PCR based detection system. So as well as by the high throughput sequencing methods, that is virome profiling, so which is carried out by IARI New Delhi. So these are the different techniques detected the presence of the viruses in Indian vineyards. So nearly 20 to 22 viruses are reported in Indian vineyards, So, but the ELISA and PCR-based detection is hardly for the few viruses. Based on the high throughput sequencing, we could detect more than 22 viruses. So 20 to 22 viruses approximately. So at ICAR-NRCG, there is a huge collection of the germplasm. So we have collected the samples from the germplasm, and we have tested the affected vineyards with the different antibodies available with us, that is grape vine leaf roll associated virus 1 and 3. So we could detect the presence of these viruses in the different germplasm plants. So and the association of the 1 and 3 is predominantly observed in different varieties. So in 6 varieties, we could record the mixed infection of the 2 viruses. So these are the total 45 germplasm we tested. So, and these are the results of the grape vine leaf roll associated virus 3. So these are the mixed infection of grape vine leaf roll associated virus 1 and 3. So as you have heard from the presentation of Dr. Sharmista, that we have initiated the Clean Plant Program. Government of India has implementing the Clean Plant Program along with the National Horticulture Board, Asian Development Bank, and ICAR. So at the present moment, these are the different steps which can be carried out in the production of. In order to manage the viral disease problems, we need to have the healthy planting material. Why we need to have the healthy planting material? Because viruses are latent, which stays throughout the life of the plant and which are affecting the productive age of the vineyards and the quality of the grapes. So we have to have the initial planting material which should be free from the viruses, so that we can increase the productive lifespan of the vineyards, and to have the quality grape yield in the grape wine. So this is the basic aim of the Clean Plant Program, which is initiated by the Government of India. So these are the basically different steps. In the initial stages, we are carrying out the hazard analysis part. So hazard analysis part is the conditions where we are collecting the samples of the grape vine from different geographical conditions, and we are testing the presence of the pathogens through high throughput sequencing testing. And based on the virome profiling, we could be able to identify the presence of the severe or presence of the severe viruses in the Indian vineyards. So based on the hazard analysis part, we can move ahead with the validation part based on the RT-PCR or ELISA based techniques. And so we can analyze the how much risk of the associated viruses in the Indian vineyard is there. So, and the maximum virus elimination based on the testing, then we will clean the plant material which is tested for the virus positive. So we will clean the plant material by using different tissue culture techniques, as you have heard from the Dr. Sharmista's lecture, that different meristem tip culture technique is there, which is helpful in eliminating the virus. There are different cryopreservation, thermotherapy. These are the combination of technologies, tissue culture technologies, which are helping to eliminate the presence of the viral pathogens. So after elimination of the viral pathogens, then it will be we will harden the plantlets and plant it into the field. So this will be the G1 block. So from G1 block, then G2, G3, and G4 blocks will be produced. So G1 block will be produced by the ICAR-NRCGrapes Pune, G2, G3, and G4 blocks, which will be developed by the nurseries. So there will be a network. It is a network project with the nurseries. So ICAR and NRCGrapes will be establishing the testing protocols for virus detection, identification, virus elimination, and development of the virus-free stocks, that is G1. So, and that G1 will be supplied to the nursery persons, and then nurseries will develop the G2, G3, and G4 blocks for subsequent, and G4 blocks will be for its further direct dissemination to the growers. So there will be a track of all the detection, and there will be subsequent detection in a year, so that we will be able to identify the which block is coming from the which mother plants, so that it will be easy to trace. So there will be a complete traceability of the production of the virus-free plantlets. So this is the overall idea of a declean plant program. We are working on the activities, different activities. So these are the different publications you can refer for those who are interested in learning the endophytic microbes as well as the viruses which were detected by the high-throughput sequencing techniques. So one of the important publication which is developed by the ICAR-NRCGrapes Pune is that package of practices for the during the fruit pruning season. So you can refer and you can all the parameters, those who are all the activities, package of practices starting from cultivation till the harvesting, all the nutrient management, water management, pest management, all the activities are listed there in this book. So you can refer. This is all about my presentation. Thank you very much for patiently listening. Thank you.
Thank you, Holkar sir. If any participants have any question, you may please ask from Holkar sir.
Hello.
Hello.
Hello. Good afternoon, Dr. Somnath.
Yes, good afternoon, ma'am.
Thank you so much for this elaborative and informative lecture.
Thank you, ma'am. Thank you.
So
Thank you.
I just want to know that I have procured fantasy seedless from NRC.
Okay.
And I observe the red coloration on the leaves in every season.
Ratna,
red
एक minute, sir.
Red
Ratna,
color.
please, एक minute, ma'am.
Then
Ratna, please.
it is like, is this a varietal character or some sort of virus? Because I have also seen the symptoms similar to the leaf roll virus.
So reddening on the leaves will be there. Is there? So I think someone is talking in between. So it is.
Okay.
That's what
Okay.
I'm saying. Arusa, ma'am, please, 2 minutes. 1 minute. Ratna, please mute yourself. Ratna.
You can send me the pictures, ma'am. We can because based on the symptoms, we can diagnose the detect the viruses associated.
So actually, what I want to ask is, do you also encounter this red coloration on the fantasy seedless leaves?
Yes, we have observed the red discoloration. So
Yes.
is there any leaf curling is there in the samples?
No,
Downward curling of the
as
leaves is
such,
there?
no, I don't think.
Okay. So we are at the
Is it?
initial stage of
It's the
validation part. So
varietal. Okay. Okay, let me see. Then I will be just also.
you
I
can send me
see
the
such
photographs,
leaves
ma'am, so
coming.
that
Okay.
it will be easy for us to understand the symptoms.
Yeah, sure.
Yes.
Sure. Just please you kindly share number.
Yeah. 7355. You can note
Thank
it
you
also.
so much.
7355.
Okay. 7355
08 6348.
08 634. Thank you so much.
Thank you, ma'am. I think no questions because most of them may be having lunch.
Yes. I think so. Is there any other questions, or we'll just end the presentation then? Sir, can we isolate endophytic microorganisms from grape plant and use it as disease control agent on same plant?
Yes. We have followed the same strategy. We have isolated the endophytic microbes from different plant parts, rather from leaves, from root, from stem, from different plant cordon, from flowering plants, from berries, we can isolate the endophytic microbes, and these endophytic microbes will be having the disease management antagonistic activity, so that that can be used for the management of different diseases in the grapes. That can be done. Thank you. How effective is chitosan spray at 2 gram per liter for controlling powdery mildew? Chitosan, we have to alternate the sprays of chitosan with the different bioformulations or different chemicals, like so that there should be a proper management of the powdery mildew. So chitosan alone will not manage the 100% manage the powdery mildew disease. So that we have to alternate the sprays with the biocontrol agents with chitosan. So alternate sprays. So will be helpful in managing the 100% managing the powdery mildew disease. So we have to do that. Both ways we can go for, either prophylactic, otherwise curative. Both the ways we can go for. But in case of the curative, so we need to go for the biocontrol as well as along with, in combination with the chemicals, because these biocontrols may not control how effective. It is again a matter of how effective is the biocontrol agents we are using, so that it is managing. So we need to alternate our sprays with the chemical fungicides for management of the curative. Any other questions?
I don't think so. There is any other question. Acha,
Is trichoderma toxic for grapes? No. One is asking, is trichoderma toxic for grape? Grapes, I did not understand the question, because sir, kindly tell us the common vectors carrying the viruses in grapes. So in grapes, most of the viruses are graft transmissible. So they are graft transmitted. For example, we have collected the cane from the infected vineyard and we have grafted on another plant or rootstock. So there is a possibility of transmitting the viruses from one plant to another through the graft transmission. Okay. So, and the vectors, if you talk about the vectors, the mealybug, for example, there are the different vectors for different diseases. For a leaf roll, grape vine leaf roll disease, mealybug is the planococcus. These planococcus species are the important vectors. Mealybugs are associated in transmitting the leaf roll disease viruses. So there are different virus vectors, mealybug, and for other group of viruses, that is sap sucking pests, that is aphids, jassids, leaf hoppers, the thrips. These are the vectors we need to identify. In India, on vector part, we have not yet worked on, so we have to identify the vectors. So first, so no work has been carried out on the vector part. So it is a total gray area of research as far as vector transmission of grape vine viruses is concerned.
Sir, are there any chitosan-based brands in market?
Brands, we would not recommend any brand, because we'll always say the chemical composition. So we will not favor any brand of the chitosan.
Should drip grafting will eliminate such menace completely?
Should tip grafting will eliminate? No. Should tip grafting will not eliminate the viral problems. So we have to go for the tissue culture techniques like meristem tip thermotherapy in combination with thermotherapy and cryotherapy, because again, meristem tip will not be 100% free from virus. So we have to go for the meristematic best way. Which form of biocontrol agent is good for long-term use? I mean, stability of agent. For biocontrol formulation, Trichoderma hargianum is widely used worldwide. Trichoderma hargianum and Trichoderma aspergilloides. So these two formulations are very effective for management of the different diseases. These formulations are working very nicely in grapevine disease management.
Any other questions?
Anyone else? I think questions are over.
Yes, sir. Thank you, sir, for your lecture. At 2:30, I suppose you'll give another lecture or Sujoy sir will give?
I will ask him because he was in different. He was in hospital, so I will confirm. If he will not be there, I will present his lecture.
Okay, so at 2:30 we all assemble again here, and sir will have another lecture. Or if sir is not there, Dr. Sujoy sir will give another his lecture. So you all
Yes.
can leave for lunch, and thank you, sir.
Thank you. Thank you. Thank you all for your patient hearing. Thank you.